7. Why DNA mutations cannot accomplish what neo-Darwinism requires. Jonathan Wells. Summary. According to neo-Darwinism, evolution takes place because of the natural selection of slight, successive variations. Some of those variations may arise through the reshuffling of existing DNA sequences, but for continuing evolution, neo-Darwinism requires that existing DNA sequences mutate into new sequences. This assumes that DNA contains a program for embryo development. DNA makes RNA makes protein makes us. Mutations in the program could then produce novel anatomical structures, and natural selection could preserve favorable ones and eliminate unfavorable ones. But DNA sequences do not even fully specify RNAs, much less proteins. And the three-dimensional arrangement of proteins in a cell requires spatial information that precedes their synthesis and is specified independently of DNA. Therefore, DNA does not contain a program for embryo development, and mutations in DNA cannot provide the raw materials for anatomical evolution. According to neo-Darwinism, living things evolve because natural selection preserves favorable variations and eliminates unfavorable ones. But natural selection cannot create new variations. Instead, these variations are supposedly generated by accidental mutations in DNA. As critics have pointed out, however, accidental mutations in DNA, like accidental changes in computer programs, are overwhelmingly harmful. The DNA sequences that code for proteins are so complex and specified, like computer code, that accidental changes in them are exceedingly unlikely to produce enough beneficial variations to supply the raw materials for evolution. The critics are right. But there is a more fundamental reason why DNA mutations are not up to the job. Evolution requires changes in the information that directs embryo development, and much of that information is independent of DNA. This chapter consists of a brief history of how neo-Darwinism came to place so much emphasis on DNA, followed by a survey of the evidence showing why that emphasis is biologically unjustified, and finally by a discussion of the implications for neo-Darwinian theory. Historical background. When Charles Darwin proposed his theory of evolution in 1859, the theory required a mechanism for organisms to transmit characteristics to their offspring. But Darwin knew of no such mechanism. In 1868 he proposed a theory of pangenesis, in which gemmules throughout the body are supposedly carried in the bloodstream to the reproductive organs, where they blend together in the sperm or egg cells to be transmitted to the next generation. But this was mere speculation, there was no evidence to support it. The Secret of Life? In 1865, Austrian monk Gregor Mendel announced the results of some hybridization experiments using pea plants. Mendel studied the inheritance of 7 traits. Each came in 2 forms, and Mendel concluded from their patterns of inheritance that one form of each trait was inherited through the male germ cell, pollen, and the other through the female germ cell, ovule. He also concluded that each trait was inherited independently of the other traits, rather than being blended together. Mendel's work was ignored for decades, until 3 European scientists took note of it in 1900. In 1905, William Bateson named the study of inheritance genetics. Soon afterwards, American biologist Walter Sutton and German biologist Theodore Boveri pointed out that during cell division the behavior of chromosomes, thread-like structures in the cell, paralleled the patterns Mendel had observed. Sutton wrote in 1902 that chromosomes may constitute the physical basis of the Mendelian law of heredity. In 1909, Danish botanist Wilhelm Johansen named Mendel's hereditary factors genes. Johansen also distinguished between phenotype, the observable properties of an individual organism, and genotype, the stable underlying biological type. Johansen and Bateson regarded Mendelian genes as abstractions rather than as material entities in chromosomes. Nevertheless, biologists continued to develop the chromosomal theory of heredity. In 1915, American biologists Thomas Hunt Morgan, Alfred Sturtevant, Hermann Müller, and Calvin Bridges wrote that, although Mendel's theory dealt with hereditary factors in an abstract way, the chromosomes furnish exactly the kind of mechanism that Mendel's theory calls for. Morgan and his colleagues also studied spontaneous changes in the hereditary factors of fruit flies. Adopting a term first introduced by Dutch botanist Hugo de Vries in a different context, Morgan and his colleagues called those spontaneous changes mutations. By this time, chromosomes were known to contain two kinds of molecules, protein and deoxyribonucleic acid, DNA. Since proteins contain 22 subunits, called amino acids, while DNA contains only 4, called nucleotides, biologists assumed that DNA was too simple to be the hereditary material and that genes were made of protein. In 1928, British bacteriologist Frederick Griffith experimented with two strains of pneumonia bacteria, one that actually causes the disease and another that doesn't. He discovered that a substance extracted from the former could turn the latter into the disease-causing strain, and he called this substance the transforming principle. It seemed that the transforming principle might be the substance of heredity. Then, in 1944, geneticists Oswald Avery, Colin MacLeod, and Macklin McCarty demonstrated experimentally that the transforming principle was not protein, but DNA. The race was on to discover the structure of DNA and how it could account for heredity. A nucleotide consists of a phosphate molecule bonded to one of 4 sugars, hence 4 subunits, called bases, and abbreviated A, T, C, and G. The phosphate molecules bond to each other, producing a long chain. By the 1950s, X-ray diffraction was being used to study crystallized DNA in order to determine its molecular structure. At the California Institute of Technology, Linus Pauling relied on X-ray diffraction studies of DNA to produce a model of DNA as a triple helix, 3 linear molecules winding around each other in a spiral pattern, with the phosphate chains on the inside. Meanwhile, James Watson and Francis Crick worked on the same problem at the Cavendish Laboratory in Cambridge, England. Watson and Crick had better X-ray diffraction data than Pauling, thanks to the work of Rosalind Franklin and John Wilkins at King's College, London. It was clear to Watson and Crick that Pauling's model was incorrect, and they modeled DNA as a double helix with the phosphate chains on the outside and the 4 bases on the inside. The characteristics of the 4 bases suggested that an A on one chain would pair preferentially with a T on the other chain, while a C on one chain would pair with a G on the other. This preferential pairing explained certain previously reported characteristics of DNA. Watson and Crick concluded that they had discovered the chemical structure of the hereditary material. On Saturday, February 28, 1953, they went to celebrate over drinks at a nearby pub, where Crick announced, "We have discovered the secret of life. B. The central dogma." When they published their model a few months later, Watson and Crick noted that it suggests a possible copying mechanism for the genetic material. A month after that they elaborated on this. Our model for deoxyribonucleic acid is, in effect, a pair of templates, each of which is complementary to the other. We imagine that prior to duplication, the 2 chains unwind and separate. Each chain then acts as a template for the formation onto itself for a new companion chain, so that eventually we shall have 2 pairs of chains, where we only had one before. Moreover, the sequence of the pairs of bases will have been duplicated exactly. In 1958, molecular biologists Matthew Meselson and Franklin Stahl published experimental evidence that DNA molecules duplicate just as Watson and Crick imagined. In keeping with the growing tendency to treat heredity materialistically, the genotype, which for Johansen was immaterial, came to mean the whole of an organism's DNA. But if the genotype consisted of DNA, how would it generate the phenotype? In the 1940s, geneticists George Beadle and Edward Tatum had shown experimentally that mutations in a single DNA sequence deprived cells of a particular enzyme. They concluded that genes act by determining the specificities of enzyme proteins. Their collaborator Norman Horowitz dubbed this the one gene, one enzyme hypothesis, later generalized to one gene, one protein, or one part of a protein. In 1955, Romanian biologist George Palade discovered microscopic particles in the cell that were later named ribosomes. Palade and others subsequently showed that ribosomes were the sites of protein synthesis in cells. In 1956, Elliot Volkin and Lazarus Astrakhan discovered what they called DNA-like ribonucleic acid, RNA, which served as an intermediate between DNA and protein synthesis. French molecular biologists François Jacob and Jacques Monod would later call the DNA-like RNA messenger RNA. In 1958, Francis Crick proposed that the specificity of a DNA segment lies solely in its nucleotide sequence, which encodes the nucleotide sequence of a molecule of messenger RNA, which in turn serves as a template for the amino acid sequence of a protein. Crick also proposed that the information encoded in DNA sequences can be transferred from DNA to protein, but not back again. He called the former the sequence hypothesis, and the latter the central dogma of molecular biology. Many writers since then, however, have used central dogma to refer to the two proposals together, a practice that is followed in the remainder of this chapter. As this brief history suggests, there is abundant evidence that DNA carries information encoded in sequences of its 4 subunits, that the coded information is transcribed into messenger RNAs, and that messenger RNAs are then translated into proteins. That is, in at least some cases. This evidence has led many people to believe that DNA thereby contains a genetic program that specifies the principal features of an organism. According to a 1987 college textbook co-authored by James Watson, we know that the instructions for how the egg develops into an adult are written in the linear sequence of bases along the DNA. Biologist Francis Collins, appointed in 2009 to be the director of the U.S. National Institutes of Health, wrote in 2006 that our DNA is an amazing script, carrying within it all of the instructions for building a human being. So the central dogma, simply stated, is, DNA makes RNA makes protein. Makes us. There have been many critics of the central dogma. One prominent critic is historian of biology Evelyn Fox Keller, who wrote in 2000. The informational content of DNA remains essential. Without it, development, life itself, cannot proceed. But current research in a number of biological disciplines has begun to put considerable pressure on biologists to reconceptualize the program for development as something considerably more complex than a set of instructions written in the alphabet of nucleotides. Nevertheless, the central dogma has become very popular. One reason is that it is consistent with neo-Darwinian theory. As Jacques Monod once said, with the central dogma, and, with, the understanding of the random physical basis of mutation that molecular biology has also provided, the mechanism of Darwinism is at last securely founded. And man has to understand that he is a mere accident. See epigenetics. Yet all biologists, even those who believe in the central dogma, know that the path from the genotype to the phenotype is affected by other factors. The Greek word epi means above, on, or in addition to. In 1942, British biologist Conrad Waddington coined the word epigenetics to mean the study of the processes involved in the mechanism by which the genes of the genotype bring about phenotypic effects. 3 years earlier, however, Waddington had used the word epigenotype to refer more broadly to the set of organizers and organizing relations to which a certain piece of tissue will be subject during its development. So from the beginning, epigenetics has had more than one meaning. In a narrow sense, it refers to the mechanisms by which DNA produces phenotypic effects. In a broad sense, it refers to all of the factors involved in development, one of which is DNA. Most biologists now use epigenetics in its narrow sense, to refer to heritable changes in a chromosome that do not alter the DNA sequence. In 2007, biologist Julie Kiefer wrote, "Epigenetics is the study of heritable changes in gene function that occur independently of alterations to primary DNA sequence. The best-studied epigenetic modifications are DNA methylation, and changes in chromatin structure by histone modifications, and histone exchange. In DNA methylation, a methyl group consisting of a carbon atom and 3 hydrogen atoms bonds chemically to a base in DNA and thereby affects whether that stretch of DNA is transcribed. Chromatin is the combination of DNA and proteins that makes up a chromosome, and the major proteins are called histones. Histones compact the long DNA molecule by serving as spools around which DNA winds. Several different molecules can modify histones by binding chemically to them, thereby changing chromosome structure and affecting DNA transcription. Furthermore, there are several kinds of histones with different properties, and molecular chaperones can exchange some for others. DNA methylation, histone modification, and histone exchange help to determine when and where specific parts of a DNA sequence are transcribed into RNA. But epigenetics in this narrow sense leaves the central dogma intact. Some biologists, however, continue to understand epigenetics more broadly. In 1993, Susan Herring wrote, "Broadly speaking, epigenetics refers to the entire series of interactions among cells and cell products that leads to embryo development." In 2002, Eva Jablonka and Marian Lam wrote that epigenetics is primarily concerned with the mechanisms through which cells become committed to a particular form or function. They concluded, recognizing that there are epigenetic inheritance systems through which non-DNA variations can be transmitted in cell and organismal lineages broadens the concept of heredity and challenges the widely accepted gene-centered neo-Darwinian version of Darwinism. The broader version of epigenetics implies that there are layers of information in an organism that are independent of DNA sequences. And indeed there are. 2. Why the central dogma fails. According to the central dogma, DNA specifies messenger RNA specify proteins, and proteins specify the organism. Yet many messenger RNAs are not completely specified by DNA sequences, and many proteins are not completely specified by messenger RNAs. DNA does not specify many RNAs. After a DNA sequence is transcribed into RNA, several processes can modify the RNA so that it does not match the original transcript. 2 well-studied processes are RNA splicing and RNA editing. RNA splicing. In plant and animal cells, most DNA sequences that code for proteins are interrupted by stretches of DNA that do not code for protein. The former are called exons, and the latter are called introns. When DNA is transcribed, exons and introns are included in the messenger RNA that will serve as the template for a protein, but the introns are then cut out and the exons are spliced back together. But one or more of the exons can be duplicated or deleted, so the final messenger RNA has a sequence that no longer corresponds to the original DNA sequence. This is called alternative splicing, and with it cells can make thousands more messenger RNAs and proteins than are encoded in DNA sequences. One DNA sequence in fruit flies generates more than 18,000 different proteins through alternative splicing. In 2010, a team of molecular biologists reported that messenger RNA transcripts from approximately 95% of multi-exon human genes are spliced in more than one way, and in most cases the resulting transcripts are variably expressed between different cell and tissue types. The diversity produced by alternative splicing has functional consequences. Among other things, alternative splicing regulates physiological changes and nerve development, and it produces enormous diversity in membrane proteins. RNA editing. In addition to alternative splicing, many animal transcripts undergo RNA editing, which can modify existing nucleotides or insert additional ones. The editing of a messenger RNA can alter the amino acid sequence of the protein it encodes, so that the protein differs from what the DNA sequence would have specified. Recent studies have demonstrated extensive RNA editing in humans. RNA editing, like RNA splicing, has functional consequences. It is widespread in animal nervous systems, where it modifies messenger RNAs of proteins involved in nerve signaling. In mice, RNA editing is necessary for the survival of stem cells that generate mature blood cells. A particularly interesting example of functional editing is found in 2 species of octopus, one living in the Antarctic and the other in the Caribbean. Both species possess very similar DNA sequences for a potassium channel in their cell membranes, but in the Antarctic octopus RNA editing changes a nucleotide in the channel's pore so that it can function better in the extreme cold. Messenger RNAs do not specify the final shape of many proteins. The function of a protein molecule depends on its 3-dimensional shape, which is determined by how the linear molecule folds. Although the amino acid sequence, which is specified by the messenger RNA sequence, often determines the final folded shape, there are many cases in which it does not. Many factors in the cell, including the degree of molecular crowding, can affect the way a protein folds. Some proteins adopt similar folded forms despite having very different amino acid sequences. Others assume different forms despite having the same or very similar amino acid sequences. Some well-known examples of the latter are prions, misfolded proteins that are pathogenic and heritable. But there are also non-pathogenic proteins in which the same amino acid sequence can fold into more than one shape. These are known as metamorphic proteins. Some proteins are intrinsically disordered. Approximately 20 to 30% of proteins in mammalian cells are inherently devoid of any ordered 3-dimensional structure. They adopt folded conformations only after interacting with other molecules. Intrinsically disordered proteins play crucial roles in regulating membrane channels and serving as hubs in intracellular signaling networks. See, most proteins are modified by glycosylation. Finally, most plant and animal proteins are chemically modified after translation by a process called glycosylation, which involves the addition of glycans. Glycans, derived from the Greek word for sweet, are composed of many small sugars chemically bonded to each other, and they can be extremely complex. In living cells, nucleotides in DNA are linked to each other in linear chains. With some exceptions, the same is true for amino acids in proteins. But sugars can be linked to each other in multiple ways. Glucose molecules, for example, can bond to other sugars at 6 different places. As a result, sugars can form branching chains that are far more elaborate than linear chains of nucleotides or amino acids. Consider molecules containing only 6 subunits, while 4 nucleotides can be linked together in various ways to form a maximum of about 4,000 different molecules, and 22 amino acids can be linked together to form a maximum of about 100 million different molecules. The dozen or so simple sugars commonly found in living cells can theoretically form more than 1 trillion different molecules. Clearly, the information-carrying capacity of the glycans in a cell far exceeds the combined capacities of the DNA and the proteins. In glycosylation, a glycan is first attached to a protein during or after its synthesis. Then complex enzymatic networks, involving dozens of interacting proteins, modify the glycans depending on the needs of the cell. The resulting modifications enable the cell to adapt to a given environment or configure it for a specific stage in embryo development. The final glycosylated form of a protein is thus very far removed from direct specification by a DNA sequence. As various biochemists have put it, the modification of proteins through enzymatic glycosylation is an event that reaches beyond the genome. It provides an additional level of information content in biological systems. Indeed, it may even pave the way for a quantum mechanics of biology, and a scientific revolution analogous to the one which transformed the field of physics in the early 20th century. So the first 2 steps of the central dogma fail. DNA does not completely specify messenger RNAs, and messenger RNAs do not completely specify proteins. But the central dogma fails most conclusively in its third and final step: proteins do not specify an organism. 3. The need for spatial information. After RNAs and proteins are synthesized in a cell, many of them must be transported to specific locations in order to function properly. In addition to their protein coding regions, some messenger RNAs have sequences called zip codes that specify the address in the cell to which they are to be transported. Like a postal zip code, however, a messenger RNA zip code is meaningless unless it matches a pre-existing address. Cells with nuclei contain microscopic fibers called microtubules. Molecular motors travel along the microtubules, transporting various cargoes throughout the cell. Using the postal code metaphor, the molecular motors could be likened to delivery trucks and the microtubules could be likened to a highway system. But destinations for intracellular transport, like the geographical addresses in a postal delivery system, must be specified independently of the cargoes. In some cases, destinations might be specified by the spatial arrangement of microtubules. In the postal metaphor, cargoes could be dispatched on a particular highway, then carried to the end of the road and simply dropped off. In other cases, destinations are known to be specified by molecules embedded in the cell membrane. Scientists originally thought that proteins could diffuse freely in a membrane, like boats floating in the sea. It is now known, however, that many membrane proteins are arranged in non-random patterns that can be quite stable. In other words, biological membranes carry spatial information. That information is mediated, in part, by a sugar code and a bioelectric code. The sugar code. As we saw above, the information-carrying capacity of branched glycans far exceeds the information-carrying capacity of DNA and proteins. There are also long and branched glycans called glycosaminoglycans, GAGs, which occur primarily on the surface of a cell. Although they are unbranched, GAGs can be assembled from dozens of different subunits, and sulfate groups can be attached to them in a wide variety of patterns. This makes GAGs some of the most information-dense molecules in nature. Indeed, the information in branched and unbranched glycans has been called the sugar code. Biologist Ronald Schnarr wrote in 1985 that there is a code on the surface of each cell that specifies its function and directs its interactions with other cells. Schnarr suspected that sugars serve as the code's alphabet. We now know that he was right. The surfaces of all living cells studied to date are covered with glycans. Cell surface glycans in early embryos of worms, fruit flies, fish, chicks, and mice change in a highly ordered manner, forming patterns corresponding to specific stages of development. They are involved in cell orientation, migration, adhesion, responses to regulatory factors, and cell-cell communication. The sugar code is interpreted by proteins called lectins, which recognize specific three-dimensional structures of glycans by means of carbohydrate recognition domains. Lectins specify a wide variety of interactions among cells. B. The bioelectric code. The cell surface code includes more than the sugar code. It has long been known that probably all living cells, not just nerve and muscle cells, generate electric fields across their membranes. In animal cells, a sodium-potassium pump in the membrane moves 3 sodium ions out of the cell while taking in 2 potassium ions. This raises the intracellular concentration of potassium ions, which then flow out of the cell through ion-selective channels in the membrane. The combined action of sodium-potassium pumps and potassium leak channels makes the interior of the cell electrically negative with respect to the exterior. The result is an endogenous electric field. The patterns of ion channels in membranes determine the forms of endogenous electric fields, which change in the course of embryo development. The endogenous electric field of a frog egg drives an ionic current through the egg. As the fertilized egg divides, and its cells rearrange themselves to form a tadpole, the electric field changes in organized ways. The same is true of chick embryos. In frog, chick, and mouse embryos, the future sites of limb development are marked by strong outward currents before the limbs appear. How do endogenous electric fields influence embryo development? One way is probably by directing cell movements. Biologists have applied artificial electric fields of physiological strength to embryonic cells and observed that some types of cells migrated toward one pole or the other. Electric fields of physiological strength can also affect cell division. Strong evidence that endogenous electric fields play significant roles in embryo development comes from artificially disrupting them and then observing the effects. Reducing an ion current associated with tail development in chick embryos results in tail abnormalities. Manipulating ion channels that generate an electric field associated with eye patterning in frog embryos results in deformed or missing eyes, or tadpoles with eyes located on the side or tail. So endogenous electric fields are certainly involved in embryo development. Reisha and Richard Borgens reviewed the experimental evidence in 1995 and concluded that such fields may provide a three-dimensional coordinate system that helps to specify form in embryos. Michael Levin argues that endogenous electric fields may function as templates of shape, and it is likely that a full understanding of development and its informational content will need to involve cracking the bioelectric code. 4. Implications for neo-Darwinism. So membrane patterns carry essential biological information. Yet that information cannot be reduced to sequence information in DNA. Even if DNA sequences completely specified messenger RNAs and proteins, the spatial distribution of proteins in the cell membrane must be specified independently, like addresses in the postal metaphor. So, what specifies membrane patterns? A. Membrane heredity. Some membrane patterns are templated by the membranes from which they are derived in the course of division. In such cases, proteins from the cell interior are incorporated during membrane growth only if they match the existing matrix. Biologist Robert Poynton proposed that biological membranes carry spatial memory, the units of which are spatially localized multisubunit proteins. As a membrane grows in preparation for division, the proteins dissociate into their subunits. Newly synthesized subunits in the cytoplasm then associate with the older subunits to form hybrid proteins that are chemically identical to the original proteins and preserve the original spatial pattern. Poynton concluded, realizing that genetic memory is one-dimensional, along a DNA molecule, where a spatial memory is likely to be two-dimensional, along membrane surfaces, and three-dimensional within the cellular interior, it is probable that spatial memory is more complicated and diverse than genetic memory. Subsequent research has found evidence consistent with Poynton's hypothesis. Cell surface templating has been well studied in single-celled organisms called protozoa. Some protozoa are covered with cilia, tiny hairs on the cell surface that beat rhythmically for locomotion and feeding. More than 50 years ago, researchers discovered that if they artificially altered the pattern of cilia, the offspring of a dividing cell could inherit the altered pattern even though the DNA was unchanged. Since then, ciliate protozoa with artificially altered patterns have been stably maintained in laboratories for thousands of generations. Stephen Ng and Joseph Frankel studied this phenomenon and concluded in 1977, the cell as an architect thus not only makes use of the genomic information to produce the appropriate building blocks, but, in addition, also arranges the building blocks according to the blueprint as defined in the pre-existing architecture. Such templating is not limited to protozoa. In 1990, Michael Locke studied patterns in caterpillar epidermis cells and concluded that their inheritance requires more than number and kind of molecule. The duplication of pattern involves relative position and orientation, factors that cannot be specified only by a base sequence. Cells with nuclei contain many different types of membranes, which Thomas Cavalier-Smith has collectively designated the membranome. Each membrane is characterized not only by its chemistry but also by its surface pattern. Cavalier-Smith called membranes that arise by growth and division of the same type genetic membranes. If any type of genetic membrane were lost, it could probably not be regenerated from its constituent molecules, even if all of the DNA sequences encoding its proteins remained, because the requisite spatial pattern would be gone. Thus, according to Cavalier-Smith, the idea that the genome contains all the information needed to make an organism is simply false. Membrane heredity, by providing chemically specific two-dimensional surfaces with mutually conserved topological relationships in the three spatial dimensions, plays a key role in the mechanisms that convert the linear information of DNA into the three-dimensional shapes of single cells and multicellular organisms. Animal development creates a complex three-dimensional multicellular organism not by starting from the linear information in DNA, but always starting from an already highly complex three-dimensional unicellular organism, the fertilized egg. But membrane heredity cannot be the whole story. In the course of embryo development, most cells do not simply duplicate the membrane patterns of the cells from which they are derived. Membrane patterns are very different in nerve cells, muscle cells, blood cells, bone cells, and liver cells, to name only a few, so membrane patterns have to change during development. The changes are not haphazard, however, they are highly regulated to generate the integrated organ systems of the adult. If membrane patterns are not specified by DNA sequences or by membrane patterns in the cells from which they are derived, how are they specified? B. A mathematical approach. The short answer is, we don't know yet. But theoretical biologists have been addressing the problem mathematically. In the 1950s, Nicholas Reshevsky and Robert Rosen introduced a new approach to the study of living things that they called relational biology. Instead of giving ontological priority to molecules, such as DNA, relational biology, as the name implies, gives ontological priority to the relations that constitute an organized system. To make their approach rigorous, Reshevsky and Rosen turned to a branch of mathematics called category theory that had been developed in the 1940s. A generalization of classical set theory, category theory can be used to model mathematically the transformations of information that occur in living things. The theory has subsequently been extended by others, including Rosen student Aloysius Louis and systems biologist Richard Sternberg. Category theory has been applied to cognitive neuroscience by Stephen Phillips and his colleagues and by André Erisman and Jamie Gomez Ramirez, though whether relational biology and category theory will succeed in explaining transformations during embryo development is not yet clear. What is clear, however, is that neo-Darwinism fails to explain evolution. See why neo-Darwinism fails. According to neo-Darwinian theory, DNA mutations supply the raw materials for evolution. If the central dogma were true, this might be the case. But the dogma that DNA makes RNA makes protein makes us fails at every step. Many RNAs are modified to the point where they no longer reflect the base sequence of the DNA from which they were transcribed. And the final folded shape of a protein, on which its function depends, is in some cases not fully specified by the messenger RNA from which it is translated. Furthermore, most proteins are extensively modified by glycosylation, the addition of complex sugars by networks of enzymes. So the final products of transcription and translation are usually very far from being specified by DNA sequences. An even more serious problem for the central dogma is that proteins must be properly localized in a cell in order for them to function properly. But DNA does not specify spatial coordinates. Instead, they are specified by the architecture of the cell and by patterns in the membrane that precede transcription and translation. So even if DNA completely specified the final form of proteins, there must still be information that is independent of the DNA. It is true that some DNA mutations can be beneficial to an organism in a particular environment. For example, the antibiotic streptomycin poisons tuberculosis bacteria by targeting their ribosomes and thereby blocking protein synthesis, but DNA mutations that slightly damage the ribosomes can prevent the antibiotic from recognizing them. Although the mutant bacteria are damaged. In the presence of the antibiotic, they are better off than undamaged bacteria. But damaging a ribosome is very different from producing the beneficial new variations in embryo development that could supply raw materials for evolution. Biologists have searched systematically for mutations that affect the development of fruit flies, roundworms, zebrafish, and mice. The effects of such mutations always fall into one of three categories. Either the embryo manages to overcome the effect of a mutation and develops normally, or the embryo is deformed, often in grotesque ways, or the embryo dies. So to judge from the available evidence, mutating the DNA of a fruit fly embryo leads to only three possible outcomes: a normal fruit fly, a defective fruit fly, or a dead fruit fly. Hardly the raw materials for evolution. 8. Theistic evolution and the extended evolutionary synthesis, does it work? Stephen C. Meyer, and K. Geiger, and Paul A. Nelson. Summary. For nearly 2 decades, many evolutionary biologists have been working to formulate new theories of evolution, in part because of the recognition that neo-Darwinian mechanisms cannot explain the origin of living things. These new ideas supposedly have more creative power than mutation and natural selection alone. This chapter will examine these new evolutionary theories and mechanisms and will show that the so-called extended synthesis has also not succeeded because it does not account for the origin of biological form and information. The extended synthesis leaves unanswered many of the same problems as neo-Darwinism and raises the same question to theistic evolutionists. Why insist on synthesizing Christian theology, or a biblical understanding of creation, with a scientifically failing theory of origins? Introduction. Charles Darwin knew that his young and ambitious theory of 1859 needed help. By the time the 6th edition of The Origin of Species was published in 1872, Darwin had gone far beyond the elegant simplicity of the processes of random variation and natural selection. So extensively did Darwin modify his theory, in fact, that the 6th edition contained many hundreds of entirely new sentences, making the last edition nearly a third again as long as the first. The project of reforming evolutionary theory, started by Darwin himself, has become an ongoing, even flourishing, industry. Throughout the 20th century, and now into the 21st, biologists have proposed one revision after another to Darwin's core concepts of randomly arising variations being sifted by natural selection. Consider one such proposed revision. In October 2014, the science journal Nature featured a print debate under the title, "Does Evolutionary Theory Need a Rethink?" with 8 prominent evolutionary biologists answering in the affirmative, and calling for an extended evolutionary synthesis, EES, to remedy the shortcomings of standard neo-Darwinian theory. Elsewhere in this volume, we and other authors have explained many of those shortcomings. Could the new EES, however, overcome the problems with the standard theory? Moreover, the skeptical scientists writing in the issue of Nature mentioned above are by no means the only scientists doubting the creative power of the mutation selection mechanism. At a recent, November 2016, conference of the Royal Society of London, called largely to address problems in the standard theory, Austrian evolutionary biologist Gerd Müller began the conference by outlining the explanatory deficits of neo-Darwinism, including its inability to explain the origin of phenotypic complexity and the origin of phenotypic novelties. He and co-author Stuart Newman have elsewhere argued that neo-Darwinism has no theory of the generative. Other biologists have explained that mutation and selection can account for the survival, but not the arrival of the fittest, that is, minor but not major changes. Accordingly, many evolutionary biologists, including many present at the Royal Society Conference, are now attempting to develop new theories of evolution based on various newly proposed evolutionary mechanisms as part of an extended evolutionary synthesis. So does this new EES solve the problems associated with the standard theory? Many theistic evolutionists have argued as much and have appealed to the hopeful promise of the EES. After acknowledging weaknesses in the standard theory, Wheaton College philosopher of science and theistic evolutionist Robert Bishop argues that, under the revised framework of the EES, while the overall picture of evolution is still one of variations filtered by natural selection, new discoveries show how developmental biology mediates between the functional biology of gene expression, cells, and anatomy, and the changes in gene frequencies of evolutionary biology. These findings will lead to a more adequate theory of evolution overall. In short, Bishop concludes, the more accurate picture of the evolutionary and developmental biology literatures is that evolutionary development and epigenetics along with other sources of genetic variation and natural selection are being forged into a new synthesis, giving us insight into how both microevolution and macroevolution happen. If Bishop is right, doubters of neo-Darwinian theory may have been hasty in passing judgment on the theory. The extended evolutionary synthesis will amend the defects of the textbook account, and materialistic or naturalistic evolutionary explanation broadly conceived will continue happily on its way. Or not. In this chapter, we examine the claims made on behalf of the EES. We show that the problems with standard evolutionary theory, or SET, an acronym devised by EES proponents themselves, persist in another form within the extended evolutionary synthesis, because those problems actually stem from the materialist foundations shared by both SET and the EES. Textbook theory and the EES are, at day's end, not so very different after all, meaning that the reforms promised by EES advocates do little or nothing to open up biological inquiry where it genuinely needs a thoroughgoing, deep, and comprehensive reformation. That such a reformation is needed is stated by EES advocates in the plainest possible language. Evolutionary biologist Adi Livnat, for instance, at the Institute of Evolution of the University of Haifa, writes bluntly of SET's explanatory failures. The modern evolutionary synthesis, SET, leaves unresolved some of the most fundamental, long-standing questions in evolutionary biology. What is the role of sex in evolution? How does complex adaptation evolve? How can selection operate effectively on genetic interactions? More recently, the molecular biology and genomics revolutions have raised a host of critical new questions, through empirical findings that the modern synthesis fails to explain, for example, the discovery of de novo genes, the immense constructive role of transposable elements in evolution, genetic variants and biochemical activity that go far beyond what traditional natural selection can maintain, perplexing cases of molecular parallelism, and more. But while many of the new mechanisms described by proponents of the extended evolutionary synthesis describe real biological phenomena, including phenomena not captured by neo-Darwinism, each of these proposed new mechanisms still fails to explain the origin of the genetic and/or epigenetic information necessary to produce new forms of animal life. Thus, the question we pose to theistic evolutionists who appropriate the neo-Darwinian mechanism will apply equally to theistic evolutionists who appropriate the EES. If the evolutionary mechanisms posited lack the creative power to produce new biological form and information, why say that these mechanisms represent God's way of creating new forms of life? 2. The three pillars of neo-Darwinism and the EES alternatives. The neo-Darwinian mechanism rests on three core claims. First, that evolutionary change occurs as the result of random, minute variations, or mutations. Second, that the process of natural selection sifts among those variations and mutations, favoring those that increase fitness, i.e., confer greater reproductive success, and eliminating those that diminish the fitness of the organisms that possess them. And third, that favored variations in the competition for survival are passed on and inherited in subsequent generations of organisms, thus causing the population to change or evolve over time. Biologists John Gehart and Mark Kushner called these three elements, variation, natural selection, and heritability, the three pillars of neo-Darwinian evolution. We shall organize our discussion of the extended evolutionary synthesis around these three key premises of the standard theory. In one or another respect, the diverse proposals made under the EES heading depart from these premises, either singly or in combination. Yet, we will argue that, in so doing, these proposals do not solve the problem of the origin of biological information, either genetic or epigenetic, necessary to build novel forms of life. To see why, let's examine several of the new mechanisms that have been proposed either to supplement or to replace the mutation-selection mechanism of standard neo-Darwinian theory. 3. New theories of evolution, the EES alternatives. A. Evolutionary developmental biology, Evo-Devo. Standard neo-Darwinian theory since the 1930s has emphasized that large-scale macroevolutionary change occurs as the inevitable byproduct of the accumulation of small-scale microevolutionary changes in gene frequencies within populations. The consensus in support of this idea began to fray within evolutionary biology during the early 1970s, when young paleontologists such as Stephen Jay Gould, Niles Eldridge, and Stephen Stanley realized that the fossil record did not show a pattern of gradual, micro-to-macro change. In 1980, at a now-famous symposium on macroevolution at the Field Museum in Chicago, the rebellion against the neo-Darwinian consensus burst into full view, exposing what developmental biologist Scott Gilbert called an underground current in evolutionary theory among theorists who had concluded that macroevolution could not be derived from microevolution. At the conference, paleontologists who doubted the micro-to-macro consensus found allies among younger developmental biologists. They were dissatisfied with neo-Darwinism in part because they knew that population genetics, the mathematical expression of neo-Darwinian theory, sought only to quantify changes in gene frequency, rather than explain either the origin of genes or novel body plans. Thus, many developmental biologists thought that neo-Darwinian theory did not offer a compelling theory of macroevolution. To formulate a more robust theory, many developmental biologists urged evolutionary theorists to incorporate insights from their discipline. And indeed, by the beginning of the 1990s, a wave of books and articles on evolutionary developmental biology, Evo-Devo, swept through the evolutionary world, proposing the idea that evolution need not proceed only by incremental small-scale mutations. Instead, Evo-Devo advocates emphasized that mutations in the genes that control animal development might have a disproportionately large effect on animal development and thus could also play a significant role in modifying animal body plans. Evo-Devo advocates thus broke ranks with classical neo-Darwinism primarily in their understanding of the size or increment of evolutionary change. Because they think that mutations that are expressed early in the development of animals are necessary to alter body plan morphogenesis, they argue that mutations must have played a significant role in generating large-scale change and, indeed, whole new animal forms during the history of life. Some Evo-Devo advocates, such as Sean Carroll and Jeffrey Schwartz, have pointed specifically to homeotic, or HOX, genes, master regulatory genes that affect the location, timing, and expression of other genes, as entities capable of producing such large-scale change. So does the Evo-Devo approach explain the large-scale changes in biological form or the origin of new animal body plans? Major but not viable, viable but not major. Despite the enthusiasm surrounding the field, evolutionary developmental biology fails, and for an obvious reason, the main proposal of the evolutionary developmental biologists, that early-acting developmental mutations can cause stable, heritable, large-scale changes in animal body plans, contradicts the results of 100 years of mutagenesis experiments on organisms such as fruit flies and nematodes, roundworms. As we saw in Chapter 2, the experiments of scientists such as Christian Nusslein-Volhard and Eric Wieschaus have shown definitively that early-acting body plan mutations invariably generate embryonic lethals, that is, dead animals incapable of further evolution. In order to generate a genuinely new body plan, of the magnitude of the difference between, say, a sea urchin and a crab, a horse or a fish, early embryonic changes must take place. Yet any mutation acting early enough to cause a substantial change in body plan always results in a dead embryo. Indeed, in their Nobel Prize-winning work, Nusslein-Volhard and Wieschaus observed that early developmental mutants never hatched as larvae. Though these mutants exhibited dramatic changes to their body plans, both in number of segments and their polarity, they were evolutionary dead ends. Dead embryos can't reproduce, and without reproduction there can be no natural selection or evolutionary change. Moreover, the same proved true of other species that have been studied, from the nematode to the mouse. Either a mutation has relatively minor effects on an animal's body plan so that it survives well enough to reproduce, or a mutation has major consequences and the animal does not survive. This generates a dilemma. Early-acting body plan mutations are eliminated immediately by natural selection because of their invariably destructive consequences. Later-acting mutations can generate viable changes in the features of animals, but these changes do not affect body plan or global animal architectures. Major change is not viable, viable change is not major. What about Hox genes? Hox, or homeotic, genes encode proteins that regulate the expression of other genes during animal development. Because they influence the pattern of development along the head-to-tail axis of the body, specifying different structures in different body segments, many Evo-Devo theorists think that Hox genes can be mutated to generate large-scale changes, macromutations, in animal form. For example, Jeffrey Schwartz at the University of Pittsburgh, in his book Sudden Origins, argues that Hox mutations may be responsible for sudden change in the fossil record. He says, "We are still in the dark about the origin of most major groups of organisms. They appear in the fossil record as Athena did from the head of Zeus, full-blown and raring to go, in contradiction to Darwin's depiction of evolution as resulting from the gradual accumulation of countless infinitesimally minute variations." His answer to this conundrum is to claim that a mutation affecting the activity of a homeobox, Hox, gene can have a profound effect, such as turning larval tunicates into the first chordates. Clearly, the potential homeobox genes have for enacting what we call evolutionary change would seem to be almost unfathomable. But there are several good reasons to doubt this claim. First, precisely because Hox genes coordinate the expression of so many other different genes, experimentally generated mutations in Hox genes have proven harmful. As McGuinness and Kurziora note, most mutations in homeotic, Hox, genes cause fatal birth defects. In fruit flies, for instance, by mutating a Hox gene, biologists have produced the dramatic Antennapedia mutant, a hapless fly with legs growing out of its head where the antennae should be. The Antennapedia mutant cannot survive in the wild, it has difficulty reproducing, and its offspring die easily. Second, Hox genes in all animal forms are expressed well after the body plan is established. In fruit flies, by the time that Hox genes are expressed, roughly 6,000 cells have already formed, and the basic geometry of the fly, its anterior and posterior and dorsal and ventral axes, is already well established. For this reason, Hox genes do not and cannot determine body plan formation. Eric Davidson and Douglas Irwin have pointed out that Hox gene expression, while necessary for correct regional or local differentiation within a body plan, for example, the refinements of limb development in both arthropods and chordates, occurs much later during embryogenesis than global body plan specification itself, which is regulated by entirely different genes. Thus, the primary origin of animal body plans is not a question of Hox gene action, but of the appearance of much deeper control elements, what Davidson calls the core developmental gene regulatory networks, DGRNs. Yet, as we saw in Chapter 2, Davidson has demonstrated that altering developmental gene regulatory networks invariably shuts down embryological animal development in animals. Third, Hox genes only provide information for building proteins that function as switches that turn other genes on and off. The genes that they regulate contain information for building proteins that form the parts of other structures and organs. The Hox genes themselves, however, do not contain information for building these structural parts. In other words, mutations in Hox genes do not have all the genetic information necessary to generate new tissues, organs, or body plans, thus, mutating them could not possibly generate new forms of animal life. Nevertheless, Jeffrey Schwartz argues that Hox mutations can explain the origin of complex structures such as the eye, as well as how organs might have been arranged to form new body plans. Yet other prominent biologists find his reasoning unconvincing for precisely the reasons just stated. In review of Schwartz's book in Nature, theoretical biologist Eeyore Sathmary, for example, notes that Hox genes don't code for the proteins out of which body parts are made. It follows, he insists, that mutations in Hox genes cannot by themselves build new body parts or body plans. Though Schwartz says he has marveled at the importance of homeobox genes in helping us to understand the basics of evolutionary change, Sathmary doubts that mutations in these genes have much creative power. After asking whether Schwartz explains the origin of new forms of life by appealing to mutations in Hox genes, Sathmary concludes, I'm afraid, he does not. To address the problem of explaining the origin of new body plans and other large-scale innovations in the history of life, Evo-Devo advocates have abandoned the idea that small, incremental variations and mutations are sufficient to produce the necessary evolutionary change. Instead, they have revived the idea of macromutations, revisiting an old idea associated with evolutionary geneticist Richard Goldschmidt and often referred to as the hopeful monster theory. Nevertheless, with this proposal a long appreciated dilemma has also re-emerged: major mutational change is not viable, while viable change is not major. Thus, evolutionary developmental biology, perhaps the most celebrated version of the extended evolutionary synthesis, has not succeeded in accounting for the origin of novel biological form. B. Self-organization. Another attempt to modify standard neo-Darwinian theory also gained prominence in the 1990s as the result of the work of a group of scientists associated with a think tank in New Mexico called the Santa Fe Institute. The scientists there developed a new theoretical approach that they called self-organization. Whereas neo-Darwinism explains the origin of biological form and structure as the consequence of natural selection acting on random mutations, self-organizational theorists suggest that biological form often arises spontaneously, or self-organizes, as a consequence of the laws of nature, or laws of form. Self-organization theorists see natural selection acting to preserve the spontaneous order that self-organizational processes have generated. They think spontaneous self-organizing order, not random genetic mutations, typically provides the ultimate source of new biological form. Thus, they especially emphasize two of the three parts of the classical neo-Darwinian triad: random mutations, and to a lesser extent, natural selection. Stuart Kauffman and the Origins of Order. One of the most well-known of the scientists associated with self-organization is a biochemist named Stuart Kauffman. In 1993 he wrote an influential book called The Origins of Order, Self-Organization and Selection in Evolution, in which he gave an incisive critique of the alleged creative power of the mutation and selection mechanism, making some criticisms similar to those described in Chapter 2 of this book. He went on to propose his own alternative theory for how new form might arise. In particular, he offered ideas for how self-organizational laws could account for the origin of the first life and the later development of the first forms of animal life during the Cambrian explosion. Kauffman says that the development of animal body plans involves two phases: morphogenesis, formation of the shape or body plan of the organism, and cell differentiation, the process by which cells become distinct types. He explores the possibility that self-organizational processes at work today during embryogenesis, specifically, in cell differentiation and body plan formation, might help explain how new animal forms originated in the past. Kauffman notes that present-day organisms' gene regulatory networks, GRNs, influence how different types of cells are differentiated during embryological development. They do this by generating predictable pathways of differentiation, patterns by which one type of cell will emerge from another over the course of embryological development, giving rise to multiple types of cells. Some become heart cells, some brain cells, and some gut cells, for example. Kauffman suggests that these pathways of differentiation may reflect self-organizing features of complex genomic regulatory networks. In other words, networks of regulatory genes in embryonic cells determine the pathways by which cells divide and differentiate. Since these patterns of cell differentiation may be determined by regulatory genes, Kauffman regards them as the inevitable byproducts of self-organizational processes. Moreover, since he asserts pathways of cell differentiation have been present in all multicellular organisms presumably since the Precambrian, he suggests that self-ordering properties inherent in a wide class of genomic regulatory networks played a significant role in the origin of the animal forms. Kauffman makes a similar case for the importance of self-organizational processes during morphogenesis. This phase involves not so much the differentiation of one cell type from another, but the arrangement and organization of different cell types into the distinct tissues and organs that jointly constitute various animal body plans. Kauffman again points to known processes of body plan development during embryogenesis and suggests that similar processes could have played an important role in the formation of the first animal body plans. He cites the importance of the structural or positional information in cells and cell membranes as the crucial determinants of how different cell types are organized into different animal forms. In Chapter 7, Jonathan Wells has discussed the importance to animal development of such epigenetic information that is outside the genome and specifies the position of the cell or membrane relative to its context. The presence of a need for such information poses a grave problem for neo-Darwinian theory. Thus, by recognizing positional information, Kaufman also rejects the neo-Darwinian assumption that a genetic program entirely determines animal development. He further regards the patterns of development that result from this positional information as evidence of self-ordering tendencies in matter and the existence of laws of biological form. Do the self-ordering processes that Kaufman cites, or laws of form, if they exist, explain the origin of animal body plans and the genetic information necessary to build them? They don't, and for two reasons. First, Kaufman's self-organizational theory does not explain the origin of the genetic information present in the genetic regulatory networks that he himself argues are necessary to cell differentiation. Instead, he begs the question as to how the information present in these regulatory networks might have originated. Indeed, though Kaufman discusses cell differentiation as a kind of self-ordering or self-organizational process, he acknowledges that the predictable pathways of differentiation that characterize this process derive from pre-existing gene regulatory networks. As Kaufman notes, the spontaneous ordering tendencies in cell differentiation are inherent in a wide class of genomic regulatory networks. Yet clearly, the genetic information in the gene regulatory networks does not come from self-ordering processes of cell differentiation. Instead, cell differentiation, to the extent that it can be properly described as self-ordering, results from pre-existing genetic sources of information. Thus, the self-organizational process that Kaufman cites cannot explain the origin of genetic information, because it derives from it, as Kaufman's own description reveals. Second, Kaufman's attempt to explain the origin of positional or epigenetic information lacks specificity and chemical plausibility. Kaufman derives the idea of positional information from a model suggested by Alan Turing in 1952. Turing proposed that the interaction of two morphogens, chemicals involved in setting up a pattern, one autocatalytic, able to independently produce copies of itself, and the other inhibitory of the autocatalytic one, could produce a differential distribution of each chemical, which would provide instructions for cell differentiation and morphogenesis. He envisioned these instructions arising independently of any pre-existing genetic material. Though Kaufman attempts to use Turing's idea to explain the origin of the positional information, he, like Turing, fails to name any specific biologic molecules that could interact in the way he envisioned so as to produce instructions capable of building a body plan. His model is entirely hypothetical. Indeed, he describes the behavior of molecules that he labels with the indistinct monikers X and Y. More importantly, Kaufman does not offer any evidence that any chemicals interacting in the way he envisions could create specific biologically relevant configurations or distributions of morphogens, apart, that is, from the processes that generate specifically arranged distributions of these chemicals in pre-existing information-rich embryonic cells today. Indeed, it is entirely implausible to think that the interaction of just two chemicals by themselves could produce enough information to coordinate all the movements and specifications that take place during embryo development. Kaufman himself acknowledges that to obtain any such order from autocatalytic reactions would require initial conditions that were incredibly fine-tuned, with high molecular specificity, and thus with a high degree of functional information from the beginning. In a later book, At Home in the Universe, Kaufman does suggest two models to demonstrate how self-ordering processes might generate positional information, but both of the models suffer from the same difficulties. First, neither is biologically realistic. One uses buttons and strings, the other flashing light to make their demonstration. Second, both rely on pre-existing information in the form of fine-tuning supplied by the computer programmer in the simulation. Kaufman himself seems to acknowledge the requirement for pre-existing information in his simulations and in biology when he says the orderliness of the cell, long attributed to the honing of Darwinian evolution, seems instead likely to arise from the dynamics of the genome network, clearly a source of pre-existing information. Order versus information. Self-organizational theorists face, in addition, a conceptual distinction that has cast doubt on the relevance of their theories to biological systems. Self-organizational theorists seek to explain the origin of order in living systems by reference to purely physical or chemical processes, or laws describing those processes. But what needs to be explained in living systems is not mainly order in the sense of simple repetitive or geometric patterns. Instead, what requires explanation is the adaptive complexity and the information, genetic and epigenetic, necessary to build it. Yet, advocates of self-organization fail to offer examples of either biological information or complex anatomical structures arising from physics and chemistry alone. Instead, they point, as Kaufman does, to embryological development unfolding predictably as the result of pre-existing information-rich gene products, cell membranes, and other cell structures. Or they offer examples of purely physical and chemical processes generating a kind of order that has little relevance to the features of living systems that most need explanation. In the latter case, self-organizational theorists often point to simple geometric shapes or repetitive forms of order arising from, or being modified by, purely physical or chemical processes as if such order provides a model for understanding the origin of biological information or body plan morphogenesis. Self-organizational theorists have cited crystals, vortices, and convection currents, or stable patterns of flashing lights, to illustrate the supposed power of physical processes to generate order for free. Crystals of salt do form as the result of forces of attraction between sodium and chloride ions. Vortices can result from gravitational and other forces acting on water in a draining bathtub. Convection currents do emerge from warm air or molten rock, rising in enclosed spaces. Nevertheless, the type of order evident in these molecules or physical systems has nothing to do with the order of arrangement. The information or specified complexity that characterizes the digital code in DNA and other higher-level information-rich biological structures. This is easiest to see in the case of the information encoded in DNA and RNA. The bases in the coding region of a section of DNA, or in an RNA transcript, are typically arranged in a non-repetitive or aperiodic way. These sections of genetic text display what information scientists call specified complexity, not simple order or redundancy. To see the difference between order and complexity, consider the difference between the following sequences. NACL 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 AZFRT MPGRT SHKLKYR. The first sequence, describing the chemical structure of salt crystals, displays what information scientists call redundancy or simple order. That's because the two constituents, Na and Cl, sodium and chloride, are highly ordered in the sense of being arranged in a simple, rigidly repetitive way. The sequence on the bottom, by contrast, exhibits complexity. In this randomly generated string of characters, there is no simple repetitive pattern. Whereas the sequence on the top could be generated by a simple rule or computer algorithm, such as, every time Na arises, attach a Cl to it, and vice versa, no rule shorter than the sequence itself could generate the bottom sequence. The information-rich sequences in DNA, RNA, and proteins, by contrast, are characterized not by either simple order or mere complexity, but instead by specified complexity. In such sequences, the irregular and unpredictable arrangement of the characters, or constituents, is also critical to the function that the sequence performs. The three sequences below illustrate these distinctions. NACL 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 NACL, order, AZFRT MPGRT SHKLKYR, complexity. Time and tide wait for no man, specified complexity. What does all this have to do with self-organization? Simply this: the law-like, self-organizing processes that generate the kind of order present in a crystal or a vortex do not also generate complex sequences or structures, still less do they generate specified complexity, the kind of order present in a gene or functionally complex organ. Laws of nature by definition describe repetitive phenomena, order, in that sense, that can be described with differential equations or universal, if-then, propositional statements. The information-bearing sequences in protein-coding DNA and RNA molecules do not exhibit such repetitive order, however. For this reason, these sequences can be neither described nor explained by reference to a natural law or law-like self-organizational process. The kind of order on display in DNA and RNA, a precise sequential order of arrangement necessary to ensure function, is not the kind that laws of nature or law-like self-organizational processes can, in principle, generate or explain. Meyer discussed this problem in his critique of teleological evolution in Chapter 6. As information theorist Hubert Yockey has explained, Attempts to relate the idea of order with biological organization or specificity must be regarded as a play on words that cannot stand careful scrutiny. Informational macromolecules can code genetic messages and therefore can carry information because the sequence of bases or residues is affected very little, if at all, by self-organizing physicochemical factors. See Michael Lynch's neutral theory of evolution. Michael Lynch, a noted evolutionary biologist at Indiana University, has proposed another EES theory to explain evolution from a non-Darwinian point of view. Interestingly, whereas in self-organizational evolutionary models, random mutation plays a relatively minor role in his account of evolutionary change, natural selection plays a minimal role. Instead, neutral, unguided, non-adaptive processes like mutation and genetic drift are the forces responsible for evolution, at least in eukaryotic organisms such as ourselves and other large multicellular organisms with relatively small populations. Why should that be? Lynch's theory is based on the equations of population genetics. These equations describe the relationship between mutation rate, natural selection, genetic drift, the effects of random chance on which organisms successfully reproduce, and population size. These equations lead to some surprising results. In particular, they suggest that, for organisms that have large population sizes such as bacteria, natural selection has the power to eliminate harmful mutations and to make universal in the population, beneficial mutations. Conversely, in large populations, the process of genetic drift, the tendency for gene variants to be lost randomly, is weak. It follows that in large populations, which would include bacteria and unicellular eukaryotic organisms, it makes sense to invoke natural selection as a significant factor in the evolution of new traits. On the other hand, the equations of population genetics also suggest that, for organisms with smaller population sizes, including most multicellular organisms and animals, natural selection will have difficulty overcoming the effects of random genetic drift, meaning that beneficial mutations are likely to be lost before they can become fixed in a population and that natural selection will not efficiently remove many slightly harmful mutations. As Lynch notes, three factors, low population sizes, low recombination rates, and high mutation rates, conspire to reduce the efficiency of natural selection with increasing organism size. Consequently, organisms in smaller populations also tend to have very large genomes and to have acquired a lot of non-coding DNA, introns, pseudogenes, and transposons. Moreover, any evolution that takes place is almost completely undirected by natural selection. It drifts neutrally without respect to adaptive advantage and has thus been called non-adaptive evolution. What does this have to do with macroevolution and the origin of new forms of animal life? Evolutionary biologists think that the ancestral groups of most new forms of animal life would likely have existed in small populations. Given the relative powerlessness of natural selection to remove extraneous DNA in such populations, Lynch argues that additional genomic elements would accumulate over time, and the genomes of animals and plants would tend to grow in size. Lynch argues that this accumulation of DNA would lead to increasing complexity in both the genome, total DNA, and phenotype, the observable characteristics, of these organisms, which would over time drive significant evolutionary change. This is his contribution to the extended evolutionary synthesis. Paradoxical aspects of Lynch's theory. Lynch has provided a powerful critique of the efficacy of the neo-Darwinian mechanism, showing mathematically that natural selection, essential to standard evolutionary theory, does not have the power to fix beneficial mutations and thus generate new traits in small populations. It does not follow from the failure of selection, however, that the accumulation of random mutations that Lynch proposes can do the job either. Without selection to drive things in a particular adaptive direction, how will near-neutral, non-adaptive processes do the job? Lynch has traded a directional process for an essentially random one. In effect, Lynch's theory attempts to explain the origin of complexity by reference to a less, not more, potent mechanism than the one offered by neo-Darwinism. Could such a paradoxical theory be true? There are several reasons to think not. First, Lynch's theory presupposes but does not explain the origin of functionally integrated cellular processes and molecular machines needed by his scenario. For example, the accumulating complexity that he envisions entails the need to edit and excise portions of messenger RNA, called introns, in order to permit its translation into proteins. An enormous molecular machine called the spliceosome does this. The problem, notes Lynch, is that introns are inside genes and get transcribed to mRNA but then have to be spliced out perfectly. If you're one nucleotide off, you get a dead transcript. Lynch recognizes, therefore, that his scenario requires the spliceosome as soon as introns entered the eukaryotic genome. So where do spliceosomes, and the information necessary to produce them, come from? Lynch doesn't say. In any case, there are good reasons to doubt that Lynch's mechanism could generate novel biological form and information. In the first place, Lynch assumes a false gene-centric view of the origin of biological form, neglecting the role that epigenetic processes play, see Chapter 7 in this book. As he writes, most of the phenotypic diversity that we perceive in the natural world is directly attributable to the peculiar structure of the eukaryotic gene. His view overlooks the crucial role of epigenetic information in the origin of animal form. Second, Lynch provides no experimental biological evidence that recombination, drift, and mutation actually produce genomic complexity. Instead, the examples he provides are entirely hypothetical. He models the growth of the gene by processes such as insertion, splicing, and domain swapping using cartoon drawings of balls, sticks, and Pac-Man-like entities. He does, of course, use various mathematical models to suggest the plausibility of the processes he invokes, but the mathematical models do not address, let alone solve, the combinatorial search problem discussed in Chapter 2 of this book. Third, though Lynch's mechanism of neutral mutational change attempts to explain the accretion of brute genomic complexity, that is, the addition of new sequences, it does not even attempt to explain functional, or specified, genomic complexity. Lynch's neutral mechanism of genomic accretion relies on purely random mutations to search the vast space of possible nucleotide and amino acid sequences that correspond to a given functional gene or section of the genome. Lynch nowhere reckons with the vast improbability of the search that his mechanism must accomplish, nor does he offer any reason to think that the probability of a successful search for functional genes or proteins would be any higher, i.e., more likely to occur, than the probability calculated in Chapter 2. He does not, therefore, solve the problem of combinatorial inflation. Instead, his model represents another version of the neutral theory of evolution that Doug Axe's experimental results about the rarity of functional proteins decisively refute, see Chapter 2. D. Neo-Lamarckian epigenetic inheritance. The third element of the neo-Darwinian triad concerns the transmission and inheritance of genetic information. Not surprisingly, the new element of the extended evolutionary synthesis we describe next questions the neo-Darwinian understanding of heredity, just as alternative theories have questioned the other aspects of the Darwinian triad: random mutation, self-organization and evolutionary developmental biology, and natural selection, near-neutral evolution. Darwin himself lacked an accurate theory of genetics that could explain how features of organisms are transmitted from one generation to the next. He thought that changes in organisms that occurred during their lifetimes from the use and disuse of different organs and anatomical systems would be transmitted to offspring through reproduction. In this respect, his theory of inheritance resembled that of an earlier evolutionary theorist named Jean-Baptiste Lamarck, who also believed in the inheritance of acquired characteristics. Lamarckian mechanisms, although unsupported by any evidence at the time, came to play an increasingly important role in Darwin's thinking, as criticisms of natural selection caused him to place more weight on the direct influence of the environment in evolutionary change. Indeed, by the 6th edition of The Origin, 1872, Darwin specifically emphasized the importance of these modes of inheritance. But with the rediscovery of Mendel's laws in 1900 and the identification of chromosomes as the material entity responsible for the transmission of inheritance, Lamarckian theories of inheritance fell out of favor. Following the rise of neo-Darwinism, the gene became the locus of all heritable change in the organism. And after 1953, biologists equated the gene with specifically arranged nucleotide bases on the DNA molecule. Recently, however, as more biologists have recognized that some biological information, epigenetic information, see Chapters 2 and 7, resides in structures outside of DNA, interest has grown in the possibility that these non-genetic sources of information may influence the course of evolution. The discovery that epigenetic information can be altered and directly inherited independently of DNA has attracted further attention to this possibility. This discovery has, in turn, led to the formulation of a contemporary neo-Lamarckian theory that that envisions changes in the non-genetic structures of an organism affecting subsequent generations during the course of evolution. Today, prominent defenders of neo-Lamarckism include Eva Jablonka of Tel Aviv University and Massimo Pigliucci of the City University of New York. Lamarck, of course, knew nothing about the role of genes, and believed that inheritance of acquired characteristics was an important driving force in evolution. Modern neo-Lamarckians, fully apprised of the reality of genetic inheritance, nevertheless think that epigenetic sources of information and structure may play some role in the evolution of biological form. According to Jablonka, neo-Lamarckism allows evolutionary possibilities denied by the modern synthesis version of evolutionary theory, which states that variations are blind, are genetic, nucleic acid-based, and that saltational events do not significantly contribute to evolutionary change. Jablonka has collected several categories of evidence in support of what she calls epigenetic inheritance systems. In the first place, she notes that in some single-celled organisms, such as E. coli and yeast, environmentally induced changes in metabolic pathways can be transmitted to the next generation independently of any changes in the cell's DNA. Second, she notes that structural information mediating organismal form and function does pass from parent to offspring independently of DNA, via membranes and other three-dimensional cellular patterns. Third, she discusses processes such as DNA methylation, a process where special enzymes attach a group of carbon and hydrogen atoms called a methyl group, CH3, to nucleotide bases within the double helix. Such processes can alter gene regulation and chromatin structure. Jablonka notes that the changes produced by these processes are often transmitted to subsequent generations of cells without any changes to the sequence of DNA. Finally, she cites a process called RNA-mediated epigenetic inheritance, a recently discovered phenomenon. Here, small RNAs, again acting in concert with special enzymes, affect gene expression and chromatin structure, and these modifications appear to be heritable independently of genes. Can any of these mechanisms help to explain the origin of animal form? Not really. By its nature, evolution requires stable, meaning permanently heritable, changes. But Jablonka's evidence shows that, where non-genetic inheritance occurs in animals, it involves structures that either a. do not change, such as membrane patterns and other persistent templates of structural information, or b. do not persist over more than several generations. Neither case generates significant evolutionary innovation in animal form. Instead, for directional evolutionary change to occur in a population of organisms, changes must be not only heritable, but permanent. Stability, the irreversible and enduring heritability of traits, is a logically inescapable requirement for any theory of evolution. This is precisely what descent with modification means. And here, Jablonka's evidence for stable non-genetic inheritance is equivocal at best, as she readily admits. Reviewing Jablonka's assembled data for animals reveals no case where an induced epigenetic change persisted permanently in any population. The heritability of such changes is transient, persisting, depending on the species in question, from a few generations up to 40. Jablonka candidly addresses this lack of evidence for stability, noting that we believe that epigenetic variants in every locus in the eukaryotic genome can be inherited, but in what manner, for how long, and under what conditions, has yet to be qualified. Consequently, despite its intriguing aspects, the evolutionary significance of neo-Lamarckian epigenetic inheritance remains uncertain, or, in Jablonka's own words, inevitably, somewhat speculative. Natural genetic engineering. University of Chicago geneticist James Shapiro has formulated another post-Darwinian EES perspective on how evolution works that he calls natural genetic engineering. Shapiro has developed an understanding of evolution that takes account of the integrated complexity of organisms as well as the importance of non-random mutations and variations in the evolutionary process. This represents a radical change to the idea of random mutations put forward by the standard evolutionary theory, and thus is another example of the EES. Shapiro observes that organisms within a population often modify themselves in response to different environmental challenges. He cites evidence showing that when populations are challenged by different environmental stresses, signals, and triggers, organisms do not generate mutations or make genetic changes randomly, that is, without respect to, or unguided by, their survival needs. Instead, populations often respond to environmental stresses or signals in a directed or regulated way. As he explains, the continued insistence on the random nature of genetic change by evolutionists should be surprising for one simple reason: empirical studies of the mutational process have inevitably discovered patterns, environmental influences, and specific biological activities at the roots of novel genetic structures and altered DNA sequences. The depth of Shapiro's challenge to orthodox neo-Darwinism can hardly be overstated. He rejects the randomness of novel variation that Darwin himself emphasized and that neo-Darwinian theorists throughout the 20th century have reaffirmed. Instead, he favors a view of the evolutionary process that emphasizes pre-programmed adaptive capacity or engineered change, where organisms respond intelligently to environmental influences, rearranging or mutating their genetic information in regulated ways to maintain viability. As an example, Shapiro notes that, contrary to the neo-Darwinian assumption that DNA alterations are accidental, all organisms possess sophisticated cellular systems for proofreading and repairing their DNA during its replication, equivalent to a quality control system in human manufacturing, where the surveillance and correction functions represent cognitive processes, rather than mechanical precision. As an example of regulated mutation, Shapiro observes that, in response to environmental assault, UV damage from sunlight, or the presence of an antibiotic, for instance, bacteria activate what is known as the SOS response system, where specialized error-prone DNA polymerases, the enzymes that copy DNA, normally left unexpressed, are synthesized and set into action, allowing the population to generate a much wider range of genetic variation than usual. The cell regulates this process using a DNA-binding protein known as LEXA that normally represses the error-prone polymerases. When the SOS system is activated by environmental damage from UV light, for example, the production of LEXA first drops dramatically, allowing expression of the error-prone polymerases. These error-prone polymerases also cause mutations, as their name implies. This characteristic is useful for increasing the mutation rate in times of environment stress, which increases the chances that some particular mutation will solve the problem for some cell or cells in the population, and allow them to survive the challenge. While at one level their error-prone role may appear counterintuitive, these mutation-generating DNA polymerases of the SOS system actually constitute essential hardware in the cell's defensive armory. From Shapiro's perspective, this survival strategy does not exemplify Darwinian randomness but rather sophisticated pre-programming, an apparatus that even the smallest cells possess to maintain viability. What's more, the carefully regulated expression of the SOS response provides evidence that cells employ the system only when needed. Shapiro argues that these and other kinds of directed, rather than random, genetic changes and responses to stimuli occur under algorithmic control. He describes the cell as a real-time distributive computer implementing various if-then subroutines. This emphatically challenges one of the three key elements of the neo-Darwinian triad, the claim that mutations and variations occur in a strictly random way. In the last 15 years, Shapiro has published a series of fascinating papers about the newly discovered capacities of cells to direct or engineer the genetic changes they need to remain viable in a range of environmental conditions. His work represents a promising avenue of new biological research, bringing new insight into how the cell's information processing system modifies and directs the expression of its genetic information in real time in response to different signals. Could it, then, also provide a solution to the problem of the origin of the information necessary to build an animal body plan? It could, except for one question that Shapiro's otherwise brilliant characterization of how organisms modify themselves doesn't address. That question? Where does the programming, the algorithmic control, that accounts for the pre-programmed adaptive capacity of living organisms come from? We know of only one source of such programming. Our uniform and repeated experience affirms that the only source for information-rich programs is intelligent agency. Or as the information theorist Henry Quasseler put it, the creation of new information is habitually associated with conscious and rational activity. Yet despite the well-established causal connection between intelligence and information-rich systems, Shapiro and other proponents of EES remain firmly committed to purely naturalistic or materialistic explanations. Stuart Kaufman, Michael Lynch, Eva Jablonka, and Charles Marshall would flatly exclude any consideration of an intelligent cause as the explanation for the origin of the information necessary to produce novel forms of life or animal body plans. Yet as we have seen, they also repeatedly presuppose the origin of information-rich systems, either genetic or epigenetic or both, without explaining how such information arose. The current situation. This same difficulty was everywhere in evidence at the Royal Society meeting in London in November of 2016. At that conference, several new mechanisms and models, including neo-Lamarckian epigenetic inheritance and natural genetic engineering, were discussed as possible remedies for the explanatory deficits of neo-Darwinism. Nevertheless, the conference failed to offer any new mechanism that could help remedy the main deficits of the neo-Darwinian synthesis, its inability to account for the origin of phenotypic novelty and, especially, the genetic and epigenetic information necessary to produce it. Of these presentations, James Shapiro's talk was clearly one of the most interesting. Shapiro reprised his case for natural genetic engineering. He presented fascinating evidence showing the non-random nature of many mutational processes that allow organisms to respond to various environmental challenges or stresses. The evidence he presented suggested, as noted above, that many organisms possess a kind of pre-programmed adaptive capacity. Yet, Shapiro again did not explain how the information inherent in such pre-programmed capacity might have originated. Other mechanisms presented at the conference betrayed similar inadequacies. For example, there was an extensive discussion of a mechanism called niche construction. Complex behaviors such as nest building by birds or dam construction by beavers represent examples of niche construction in which some organisms themselves demonstrate the capacity to alter their environment in ways that may affect the adaptation of subsequent generations to that environment. Yet no advocate of niche construction at the meeting explained how the capacity for such complex behaviors arose de novo in ancestral populations, as they must have done if the naturalistic evolutionary story is true. Rather, these complex behaviors were taken as givens, leaving the critical question of their origins more or less untouched. While there is abundant evidence that animals can learn and transmit new behaviors to their offspring, crows in Japan, for instance, have learned how to use automobile traffic to crack open nuts. All such evidence presupposes the prior existence of specific functional capacities enabling observation, learning, and the like. The evolutionary accounts of niche construction theory therefore collide repeatedly with a brick wall marked original complex functional capacity required here, without which, or beyond which, there would simply be nothing interesting to observe. Indeed, the new mechanisms offered by critics of neo-Darwinism at the Royal Society conference, whether treated as part of an extended neo-Darwinian synthesis or as the basis of a fundamentally new theory of evolution, did not attempt to explain how the information necessary for generating genuine novelty might have arisen. Instead, the mechanisms that were discussed produced at best minor microevolutionary changes such as changes in wing coloration of butterflies or the so-called polymorphisms of stickleback fishes. Moreover, the mechanisms presented there, niche construction, neo-Lamarckian epigenetic inheritance, and natural genetic engineering, for example, either presupposed the prior existence of the genetic information necessary to generate novelty, or they did not address the mystery of the origin of that information, and morphological novelty, at all. Thus, even a science reporter friendly to the EES, Susan Mazer, complained in the Huffington Post of a lack of momentousness. What was the point of attracting a distinguished international gathering if the speakers had little new science to present? Why waste everyone's time and money? 4. Conclusion, a timid reformation that leaves us where we were. The metaphor of a city bounded by a high wall helps to illuminate the situation now faced in evolutionary biology. Despite the difficulties with the standard theory that proponents of the extended evolutionary synthesis acknowledge, EES theorists remain fully neo-Darwinian in the essential sense that separates them from proponents of intelligent design or special creation, namely, their views about the ultimate sources of biological form and information. EES proponents assume that novel form and information must arise from only natural, meaning physical or material, wellsprings. It is not imparted to the biosphere by an actual purposeful intelligence, or, more precisely, we may not infer that biological information has an intelligent source, at least, not one external to the evolutionary process itself. Such inferences would in their view violate the rule of methodological naturalism, see Chapter 19. Methodological naturalism defines the walls of the city-bounding scientific explanation proper, an intelligent design, that is, real teleology involving an actual purposeful intelligence, lies beyond those walls. Thus, while the proponents of the extended evolutionary synthesis urge openness to new ideas on the part of the set establishment, and can be blistering in their critiques of textbook theory, they stay obediently within the city walls. Indeed, the walls of methodological naturalism are high and impassable, and for most evolutionary biologists, ever in sight. Most never consider leaving the boundaries defined by methodological naturalism. Hence, the problem created by accepting it, in particular, the problem of explaining the origin of information for which we know of only one cause, remains unsolved. One can accomplish only so much within those walls. But why should it be the case that the walls stand so high, when every proposal of the EES can be shown to fail, and methodological naturalism is clearly preventing consideration of other more causally adequate, plausible, and fruitful ideas? Surely someone is willing to scale the wall to see what might lie on the other side. And why, specifically, should theistic evolutionists, who already presumably believe there is something on the other side of the wall, remain behind it? 9. Evidence from embryology challenges evolutionary theory. Sheena Tyler. Summary. How does an egg develop into the distinctive body form of an elephant, as opposed to a grasshopper or a kangaroo? It remains a mystery to this day how these body forms are generated. This is a major problem for evolutionists, because their claim that the various forms of life arose by changes in a common developmental program depends on knowledge of this elusive program. This chapter will demonstrate how embryological processes exhibit the hallmarks of intelligent design rather than the tinkering of blind, random mutations required by evolutionary theory. It will also illuminate evidences from embryology that point to distinct types of life, which exhibit fundamental differences in design between them, rather than a continuous gradation of forms tracing back to a primitive common ancestor. Introduction. In 1859, Charles Darwin wrote that embryology is to me by far the strongest single class of facts in favor of change of form. To this day, embryological mechanisms are placed center stage to explain the evolutionary origin of diverse body forms. This chapter examines whether experimental evidence emerging from embryology either supports or refutes these claims. The first section investigates whether alterations to genes involved in development can generate evolutionary changes. What evidence is there for how changes in such genes might create large-scale transformations to an organism's body plan? The second section looks at whether body plans are assembled according to random and unguided Darwinian processes, or are these assemblies orchestrated, bearing hallmarks of intelligent design? The third section explores whether embryological development supports the idea of a continuous gradation of form among living things, which can all be traced back historically to a primitive common ancestor. Alternatively, does the evidence suggest the existence of distinct types in nature, with common modes of assembly, clearly distinct from neighboring types of assemblies? The experimental findings discussed here have far-reaching implications to challenge the strongest class of facts, which Darwin marshaled to support his theory. Part 1. Nature's greatest secret, the form question. 1.1 Genotype to phenotype. The body plan of an organism refers to its major structural features or form by which it can be recognized. Examples are the snail, with its familiar shell, and the sea star with its radial arms and sucker feet. The outward visible form, phenotype, is considered by most biologists to be determined by a genetic blueprint, the genotype. How this plan or form is generated remains one of the most fascinating but elusive challenges for science. This has huge implications, for it is said that we will not understand evolution until we understand how organisms are produced in development. The problem of the knowledge gap between genotype and phenotype has been well recognized, as the following quotes from various embryologists indicate, emphases added. Ray Keller, one of the enduring mysteries of developmental and cell biology is morphogenesis, the process of how the heritable body plan is shaped at every stage from the fertilized egg. Brian Goodwin, the problem of biological form remains unresolved despite the known details of gene activities in embryonic development. Franklin Harold, what orchestrates the hubbub of enzymes, filaments, membranes and polymers into a recurring pattern? We really do not know. Michael Levin, deciphering and learning to control shape is arguably the fundamental problem of biology and medicine. Beverly Purnell, transforming a single fertilized egg into a complex animal is a marvel and a mystery. Lars Hufnagel, how growth is controlled and coordinated, remain largely unanswered questions. But if the mechanisms by which the programs of development translate into form remain to be discovered, then the mechanisms underlying large-scale evolutionary change remain highly speculative, since these essentially depend on fundamental changes in the black box of the developmental program. 1.2 Historical sketch. Evidence that embryological development appears to have coordinating influences has been emerging over the past 100 years. In Germany, Hans Driesch recognized that embryo parts have the capacity to generate the whole, via these parts working harmoniously together. He proposed too that the fate, identity and destination, of cells is determined by their position, according to a reference system of fixed coordinates. In Russia, Alexander Gurwitch found evidence for a guiding field of force, the Embryonalsfeld, which ordered the whole of development throughout the embryo. In the following decades, a large body of experimental data was marshaled in support of a morphogenetic field system, which thus became the established research paradigm, integral to the quest to discover the underlying laws governing organismal form. In contrast, the American geneticist Thomas Hunt Morgan viewed the causal basis of form to reside exclusively in genes and their products, rather than in fields. Morgan's student Theodosius Dobzhansky defined evolution to be the result of changes in gene frequency, with genetics as the motor for evolution. Following the Second World War, this view rose into dominance. This was the synthetic theory, or modern synthesis, in which large-scale evolution was explained by random, undirected mutations acted on by natural selection, with an emphasis on how different permutations of genes were distributed in populations over time. However, there was a growing recognition that our knowledge of changes in genes and their products remains insufficient to explain development of form, morphogenesis. In addition, there was increasing frustration at the lack of evidence from genetics to explain large-scale evolutionary change. One of the major tenets of the modern synthesis is that of extrapolation, the notion that macroevolution, the evolution of organisms with the distinctly different body plans of higher taxonomic groups, is fully explained by microevolutionary processes that give rise to varieties within species and genera. Disagreements over this notion were so intense at a major conference on macroevolution that proceedings of the conference were never published. Paleontologists and biologists such as Stephen Jay Gould and Francisco Ayala asserted that microevolutionary events indeed do not explain macroevolutionary processes. As Gilbert, Opitz, and Raff commented, microevolution concerns only survival of the fittest, not arrival of the fittest population. Genetics must change if it is not to become irrelevant to evolution. Evolutionary developmental biology, Evo-Devo, and current trends as an attempted solution to the above unrest, evolutionary developmental biology, or Evo-Devo, emerged in the 1990s when researchers proposed that alterations to genes involved in development can explain evolutionary changes. A number of themes are embodied in this credo. These include the idea that changes in the sets of genes available to organisms for use in development, molecular toolkits, and all the pathways for exchanging information and modulating cellular behavior, signal transduction pathways, can account for macroevolution. Gould postulated another idea, that changes in the relative timing of developmental events and different growth of parts relative to each other could serve as mechanisms to rapidly create new body plans. Another proposal is the module concept, with complex organs envisaged as arising by the addition of new modules. Yet another idea is self-organization, in which forms arise spontaneously according to the laws of nature rather than from random genetic mutations. However, there are numerous problems with Evo-Devo and the above ideas. These are briefly outlined as follows. For more, see Chapter 8 on the extended evolutionary synthesis. Without knowing the steps by which the genotype is actually transformed into the phenotype, it is a large step of faith to assume how such unknown pathways would have been transformed in evolution. Indeed, it is the recognition that these transformations are such a black box that led Joan Baguna and Jordi Garcia Fernandez to urge extreme caution when such inferences are made. Examples of Evo-Devo in action fall far short of providing experimental evidence of macroevolution at work. If these processes did occur, it should be possible to create large-scale transformations to an organism's body plan. However, experiments which generate mutations in genes acting on body plans tend to produce overwhelmingly defective or lethal embryos. A mutation in the UB's gene in Drosophila, the fruit fly, leads to an extra set of wings, but these are not functional, lacking muscles that work the wings and other essential structures, and the extra wings prevent the fly from flying. Moreover, the extra wings are a mere duplication of a pre-existing structure, rather than one new to nature. The eminent developmental biologist Eric Davidson and colleagues focused for decades on gene regulatory networks, GRNs, claiming that evolutionary change in body plans results from alteration of the functional organization of the GRNs. It is interesting to see, then, some of the best evidences Davidson forwards. These include the loss of spines in sticklebacks, and loss of eyes in cavefish. However, this represents loss of information, and certainly is no evidence for the generation of novel macroevolutionary morphologies. Even Davidson admits that mutations affecting GRNs that are implicated in the development of the body plan lead to catastrophic loss of the body part, or are lethal. In other words, there is no experimental evidence that a simple switch or even a series of mutations that alter GRNs can in practice lead to a new body plan. These remain speculations, not observations. For instance, Isabel Peter and Eric Davidson imagine, as previously speculated, mobile elements could have provided a major mechanism of GRN evolution. Alterations in GRN structure are easily imagined, emphases added. Douglas Futuama concludes that some authors make hyperbolic claims for Evo-Devo well beyond what current evidence supports. Transformations from one form to another are never simple, but require completely integrated transformation of form for all associated structures, and all of these must be functional. For instance, the body plan of highly elongated fishes differs in a number of ways compared to stout-bodied close relatives. Elongate fishes tend to have longer and or more numerous vertebrae, longer bones within the skull, and longer fins. That such transformations are not simple is further indicated by the recognition that body shape indeed has a complex genetic basis. I am interested in boat design, Figure 9.1a. If in relation to the original plans a wider boat were to be constructed, these widths change along the boat's length, requiring the sides to be reformed and lengthened. Computer-aided design can make changes in boat design appear easy, Figure 9.1b-c, but this is possible only because the software was intelligently designed in the first place, to enable each part of the boat to be modified in relation to the whole. Similarly, organisms that vary their body length relative to other species must modify each part in relation to the topography of the whole organism in 3 dimensions, and this involves the coordinated addition or removal of cells in various tissues, figures 9.1d and e. The idea that evolutionary modifications can occur by simple changes in the timing of development is also popular. However, a report of molecular geneticist Professor Susan Cole's work regarding gene activity in chick development shows that, if the timing of these genes' activity does not remain tightly regulated, the tissue either will not form at all or will form with defects. When self-organizing chemistry occurs within a living cell, it comes under guidance and constraint from the cellular system as a whole. According to biochemist Franklin Harold, new gene products are never altogether at liberty, they are released into a cellular milieu that already possesses spatial structure, under the influence of the existing order. In other words, a higher order of information governs and directs molecular assembly. Part 2. Development is orchestrated. Consider the novel Mr. Stand Fast, by John Buchan. Our hero Richard Hannay is wrongly under suspicion and is being hotly pursued over the moors by half of Scotland's police force. They are closing in, not knowing that Hannay is an undercover British government agent, and the outcome of the First World War is at stake. Just in time an old friend, Sir Archie, appears, who whisks him away in his biplane to safety. In this gripping thriller, villains and heroes encounter one another in just the right times and places. We next find Hannay in a London Underground station during an air raid. He is hunting for his archenemy, a German spymaster. Of all the places in London, Hannay spots him, just there, at that very moment. One can see that the author has orchestrated all the plot points so as to accomplish his aim. If we move our spotlight from the encounters of the novel to the embryology scene, do we see the molecular players encounter each other by chance, or do we see them in just the right times and places? I will look at this question by describing some of the key aspects of morphogenesis in different regions of the body. Recurrent themes will appear, themes related to mechanisms of information storage. One theme is positional information, by which cells have their position specified in relation to a spatial coordinate system of reference. Another theme is the pre-pattern, which provides a template or scaffold for the organization of some subsequent structure. I will discuss examples of each of these methods of storing the information necessary for morphogenesis of various body systems, stressing the point that, without pre-existing information, no morphogenesis can take place. If one may generalize, a pre-pattern of some sort, perhaps deriving from some sort of even more prior positional information, directs gene expression, which in turn directs cell behavior, which in turn directs morphogenesis. There is a flow of information from the first stages of development to its outworkings in final bodily form that must be highly orchestrated and choreographed, to use words we will see repeatedly in the researcher's own language. 2.1 Skeletal development. In the development of the limb in vertebrates, an outwardly directed electric current plays a role in identifying the correct location of the future limb, Figure 9.2. See Chapter 7 in this volume. Particular genes and their products are active, producing chemical signals integrated into a four-dimensional patterning system. These chemical signals help to specify the development of the limb, Figures 8.2b and c. It is noteworthy that these signals are produced at the appropriate time and location, which implies an underlying pre-pattern that directs the gene expression itself. The size and shape of skeletal elements appear to be, according to neuroscientist Charles Kimmel, exquisitely regulated and precise. This indicates that a pre-pattern for skeletal development is likely to exist. For instance, in the zebrafish larval head, cartilage cells begin to appear at precise locations, such as where future joints will emerge. This may be guided by a pre-pattern organizing specific gene expression to this region. There is evidence too of a pre-pattern in mammalian bone development. Within the bones are lattice-like scaffolds, or trabeculae, which become highly orientated according to the future stresses on the bone. The lattice patterns appear in the embryo before any mechanical loading, suggesting they may be organized by a predetermined template. 2.2 The nervous system. Development of the central nervous system requires precise and exquisitely regulated gene expression patterns, according to neuroscientist Carla Metzer, Sosa and colleagues. For example, the brain and central nervous system arise from neural progenitor cells, NPCs, which multiply and develop into nerve cells, growing very long extensions, axons and dendrites, that become connected to target cells via synapses, junctions between nerve cells. The information that directs the connectivity is considered to arise in the NPCs from expression of a combinatorial code of transcription factors. Transcription factors are proteins that affect the expression of other genes. This combinatorial code must in turn arise and be exquisitely controlled by pre-existing information, the pre-pattern. The result is what neuroscientist and Yale University professor Daniel Colan-Ramos describes as the intricate orchestration of cell migration, axon guidance, growth of dendrites, and synapse formation of the nervous system. It must be orchestrated because synapses must connect together precisely, with 100% accuracy. In fact, what I have described only begins to illustrate the complexity and interconnectedness of neural development, and the information necessary to accomplish that development. There is a rich tapestry of informational signatures to be found both within DNA and also beyond it. For more information on this topic, see Chapter 7 in this volume. All these forms of information function like codes that cells read, integrate, and interpret to guide their connectivity with other neurons. The information can be specified as follows. Pre-patterning information. Previously it was thought that synapse formation occurred only after neighboring neurons made contact with one another. However, even prior to such contact, synaptic hotspots of specialization occur, suggesting that pre-patterning tells the neurons where synapses will form. A transcriptional code, biochemical messengers secreted along the various body axes are thought to communicate positional information to the NPCs. This causes them to produce a combinatorial code of transcription factors, which in turn provides information governing neural connectivity. Moreover, cells become specialized to become those forming sensory organs, example, the eye, requiring careful orchestration of complex regulatory mechanisms, according to developmental neurobiologist Jean-Pierre Saint-Genet, professor at the University of Toulouse. A histone code. Chromosomes are composed of strands of DNA wrapped around proteins called histones. Modification of these histones is involved in the activation or the repression of gene activity. Modifications of histone proteins provide a histone code, which multiplies the combinatorial potential and thus the collective informational capacity of the genes. Geneticists Bing Yao and Peng Jin remark that this code delicately orchestrates with extracellular cues to determine the accurate regulation of neurogenesis. A cell surface code. Cell surface carbohydrate protein complexes have a vast informational combinatorial potential. Their role is suggested because changes in their expression correlate with events during nervous system development and their inhibition perturbs this development. See Chapter 7 for more information. A bioelectric code, consisting of electrical currents in certain regions of the developing embryo. For example, there is electrical activity that predicts the subsequent patterning of the nervous system, and that interfering with the neural electrical signal leads to neural developmental defects. Once again, see Chapter 7 for more on this. Thus the DNA and various epigenetic sources beyond DNA provide coded information that is both sophisticated and integrated together in their activities, and that together help direct the formation of the nervous system. 2.3 Tooth development. Tooth development in mammals appears to be guided by positional information, which is tailor-made for each tooth. The shape of each tooth grades with its immediate neighbors, so that the dentition is integrated into a harmonious whole. Moreover, this integration extends to coordinated signaling interactions between the development of the teeth with their anchoring ligaments and the surrounding bone. By appropriate deposition and reabsorption of bone, the bony socket of the tooth thus becomes precisely molded with the developing shape of each tooth root. 2.4 Heart and circulatory system development. Development of the heart is a wondrous and precisely orchestrated series of molecular and morphogenetic events, in the view of heart disease specialist Deepak Srivastava. Heart development arises from a region of heart-forming cells in the early embryo known as the first heart field. These cells develop, migrate, and form into a linear tube, the heart tube. Later, a group of cells known as the second heart field migrate to the heart tube, enabling the heart tube to become elongated, then loop to the right, expand, and become remodeled into the heart chambers, atria and ventricles. Cells from the neural tube also migrate into the heart tube and are required for formation of the heart valves and compartments which separate oxygenated and deoxygenated blood. A very large number of genes are involved in these processes, with a network of transcription pathways activating each other's expression. Irfan Kathiria and colleagues from the University of California report how cardiac transcription factors choreograph the expression of thousands of genes at each stage of heart development, by interacting with cofactors, and by binding with a constellation of regulatory DNA elements. These networks interplay to orchestrate the sequential deployment of the cardiac gene expression program, Figure 9.3. In addition, the muscles of the heart do not contract randomly and chaotically. Rather, the atria and ventricle chambers contract sequentially, which is crucial to the effective expulsion of blood. This is facilitated by microscopic muscle cell proteins precisely registered together. The integration of these contractions globally throughout the heart is provided by specialized cells organized into an electrical wiring system. The second heart field, mentioned earlier, contributes to the development of this electrical conduction system. Electrical impulses generated from pacemaker cells spread rapidly across both atria, resulting in simultaneous contraction of the atria. They then converge on the atrioventricular node, which delays the spread of the electrical activity so that the atria contract before the ventricles. Electrical activity is then spread rapidly through the ventricles, through a specialized fast-conducting system or ventricular conduction system, leading to the sequential contraction of heart muscle, progressing from the apex to maximize the expulsion of blood. Development of this conductive system involves the combinatorial effect of numerous cardiac-specific transcription factors, implying a programmed, informational capacity to the second heart field. This results in development of the electrical system becoming integrated with the shape and form of the heart. Cardiologist Nikhil Munshi describes how even subtle perturbation of this finely orchestrated electric activation pattern can cause clinically important or fatal cardiac arrhythmias, indicating its essential importance. However, the heart also requires a circulatory system to pump blood into, and it appears that development of both the heart and circulation are simultaneously coordinated. Cells destined to become blood vessels aggregate together in islands, then migrate and connect to form a network of tubes. This provides the scaffold for development of arteries, veins, and capillaries by remodeling and subsequent vessel sprouting, angiogenesis. Various substances secreted by cells to regulate development, growth factors, orchestrate the growth of vessels in a very ordered pattern, according to cardiology researchers A. S. Chung and colleagues. There is great fidelity to this pattern, with the anatomy and branching points of major vessels being highly reproducible, precisely positioned in relation to the organs they serve, and exactly plumbed into other major vessels, with evidence that this is genetically pre-patterned. For instance, angiogenesis is initiated by vessel leakage of plasma proteins, which then form a scaffold for migrating vessel lining, endothelial, cells, guided by vascular endothelial growth factors, VEGFs, liberated in an orchestrated sequence. For the proper functioning of the cardiovascular system, arteries, veins, and lymphatics do not have a uniform morphology but have distinctly different structural features so as to perform different tasks. Arteries possess thick internal layers in order to cope with blood exiting from the heart under high pressure. Veins are under low pressure, and thin walls are sufficient, but are in addition constructed so as to allow the passage of white blood cells across them. They also possess valves for the one-way flow of blood and to prevent backflow into the capillaries. This arterial and venous (AV) specification requires the correct spatial and temporal expression of many genes orchestrated by a relatively large set of transcription factors, working together in concert. Researchers Jason Fish and Joshua Wythe similarly recognize AV specification to be a highly coordinated process involving the intersection and carefully orchestrated activity of multiple signaling cascades and transcriptional networks, including the hedgehog, VEGF, TGF, WNT, and Notch signaling pathways. The crucial role of VEGF is seen when loss of even one copy of VEGF in mice results in profound vascular defects leading to embryonic lethality. Fish and Wythe reiterate the exquisite orchestration required for these various processes to occur, sometimes simultaneously, with a plethora of signaling pathways acting to coordinate the establishment of the vasculature. Moreover, vessels develop in relation to their function and the specific needs of the organ in which they may be located. For instance, embryonic brain development is intimately associated with development of the blood vessels there. The brain microvasculature strictly minimizes permeability, while in contrast organs such as the liver or bone marrow possess a vessel structure which promotes permeability between blood cells and proteins. The development of the cardiovascular system, in relation to the functional demands of the vasculature, implies a yet even greater level of integration and crosstalk of developmental information. 2.5 The nature of information. If organisms are not intelligently designed, they must be cobbled together by non-intelligent mechanisms. Tinkering mechanisms are at the heart of modern theories of evolution. However, the above accounts indicate many evidences of an intricate orchestration of patterning mechanisms to enable the concurrent coordination of various organs and organ systems, integrated with reproductive behavior. My daughters play in a youth orchestra. During rehearsals, whenever left to themselves, the young players immediately start up their own tunes or practices. Very soon there is only noisy chaos. It requires the conductor to bring order out of this chaos, and also, intelligent information encoded in the composer's manuscript needs to be interpreted by the conductor and musicians to produce the harmonious melody. So, the recourse of various authors to the metaphor of orchestration of development is interesting, for such orchestration is a signature of intelligent causation. It is not just a genetic code, but many codes together providing a rich combinatorial capacity for information storage. To the above list of codes could also be added the splicing codes, the cytoskeleton codes, the apoptosis code, and the ubiquitin code. In 2016 a whole special issue of the journal Philosophical Transactions of the Royal Society was devoted to the theme, DNA as Information. Introducing the issue, Julian Cartwright and colleagues state that biological codes not only transmit information but also interpret it, and that the genetic code information even includes how to produce new senders and receivers. They recognize too the programming aspect, being written in language yet to be decoded, and being not merely a simple linear list of instructions, but a program with subroutines, callbacks, loops and all the complexity that implies. Thus, the premise of a simple, linear flow of information in gene sequences is giving way to a new model of development involving biological networks within hierarchical tiers, with information moving multidirectionally both within and between the tiers. The engineer and physicist Werner Gitt recognizes that technological systems, engineered objects, and works of art did not come into existence by self-organization of matter, but were preceded by establishing the required information. He adds that it has not been shown experimentally how information can arise spontaneously in matter. It is only through guidance that processes run contrary to natural law's directions, to attain a goal that unguided processes could never. Gitt has discovered that human and machine languages exhibit 5 levels of information. The lowest level of information is statistical, a symbol or number of symbols. However, the mere statistical, quantitative evaluation of a symbol sequence ignores whether or not such information is meaningful. Language systems exhibit a second level of information, comprising codes, sets of abstract symbols, combined with syntax, rules for construction and arrangement of words. Languages also possess a third level, called semantics, the meaning of the message being conveyed. Fourth, they exhibit pragmatics, action that the sender expects the receiver to perform, and a fifth, highest level, apobetics, purpose, the intended goal. Gitt coins the term universal information, UI, for systems expressing these characteristics. UI thus comprises symbolically coded messages conveying the expected actions and the intended purposes. The DNA of living things utilizes a genuine coding system, and all these 5 levels of information are observed. Gitt concludes that any code system is always the result of an intellectual process that requires an intelligent originator. This has implications for recognizing that there must be an intelligent originator of the various code systems found in embryological development. Thus, from the above accounts, the notion of macroevolution occurring through changes in genes involved in development remains a theater of dreams and speculations. So, does development reveal any story of life's history? This is our next focus. Part 3. Basic types of life. According to evolutionary theory, all organisms are historically connected in the so-called evolutionary tree, in which body forms can be traced back through gradual changes from simpler body plans, continuously grading over many generations, ultimately derived from a primitive common ancestor. A contrasting view discerns distinct kinds or types of organisms that share a basic body plan and mode of development. These distinct assemblages of organisms most closely approximate to the modern family taxonomic level. Their distinctiveness argues that each group may have a separate and unique origin. Which of these 2 scenarios the evidence better supports will now be explored. 3.1. Hybridization data. Crossbreeding, hybridization between species, is very common. For instance, 1 in 10 species of bird is known to hybridize with another species. This suggests that the eggs and sperm from the hybrid's parents must share a developmental machinery that is compatible in order to produce viable, live offspring. Therefore, probing the data to find which species and genera can be united together by hybridization is an evidence-based, i.e., empirical, method to discern whether natural groupings exist. Analysis of this data provides strong evidence that this is indeed the case. The following provide a few examples. The horse family, Equidae. All species within the horse family, horses, donkeys, asses, and zebras, are united by their ability to hybridize. But they do not hybridize with their nearest suggested evolutionary relatives, the tapirs and rhinoceri, figure 9.4. The cat family, Felicidae. Hybridization unites various species, and also several genera. For instance, the puma concolor, can hybridize with the leopard, Panthera pardus. The ruddy Abyssinian cat breed, figure 9.5a, never exhibits a spotted coat, and neither does the seal point Siamese, figure 9.5b. However, when a ruddy Abyssinian was crossed with a seal point Siamese, and the offspring was back-crossed to another Siamese, the surprising outcome was a spotted cat. This is now recognized as a new rare breed known as the ossicat, figure 8.5c, because of its similarity to the ocelot. The ossicat spots are an example of an atavism, described by Brian Hall as the reappearance of a character known in distant ancestors and not seen in the parents or recent ancestors. The bear family, Ursidae. 6 of the 8 species of bears, example, the polar bear, American black bear, Asiatic black bear, sloth bear, and sun bear, are united by hybridization. Ducks, geese, and swans, Anatidae. Remarkably, 81% of all 149 species of ducks, geese, and swans, Anatidae, hybridize with one another. Over 50% of these hybridize between genera, groups of species sharing similar features. Yet there are no known hybrids between the Anatidae and members of any neighboring taxonomic groups. 20% of all known hybrids are fertile, suggesting that the Anatidae are genetically related but clearly distinguished from other taxonomic groups, taxa, of birds. Cranes, Gruidae. Hybridization links 14 of the 15 species and all 4 genera of cranes together within the family. These also link the 2 subfamilies, crowned cranes and true cranes. Pelicans, Pelicanidae. 6 out of 7 pelican species can hybridize, but hybridization between pelicans and any other proposed relatives is unknown. Storks, Cichonidae. 7 of the 19 species of storks hybridize, but no hybridization is found between storks and other bird families. Siegfried Scherer and colleagues demarcated a number of groups by such interspecific hybridization, which they designated as basic types. 2 individuals were viewed to belong to the same basic type if they hybridized with each other, or if they hybridized with a third organism in common. At least 72 basic types have been identified, with the prospects of many more from untapped data. Most commonly, the basic types are identifiable with the family, subfamily, or superfamily taxonomic level. A number of findings emerge from these analyses. Hybridization is common within basic types, but absent between them, indicating natural gaps or divisions between the basic type and their nearest neighbors. The basic types distinguished by hybridization strongly correlate with the outward form or morphology of the group. Importantly, this is exactly as would be predicted if the members of a basic type shared a developmental machinery, which generates their common form. Within a basic type, although the basic form is shared between members, hybridization also links together species with relatively divergent morphology. For instance, hybridization unites various species of parrots, which nonetheless still share the recognizably parrot-like form. This suggests a certain developmental capacity for variation or plasticity within the basic type. Evidence of atavisms within some basic types indicates a hidden ancestral reservoir of genetic potential. Indeed, Brian Hall considers atavisms to be the visible expression of huge hidden potential for change in form with which all organisms are endowed. This hints at the notion of genetically complex ancestors. Genetic mechanisms are known to exist by which this ancestral reservoir is considered to generate new variations in form following hybridization. This is not a concession to microevolution, which refers to incremental emergence of novelty on an evolutionary trajectory. Rather, the appearance of novel traits represents diversification from a pre-existing, information-rich genotype. 3.2 Cleavage pattern. The spatial arrangement of cells within an embryo as they divide is known as the cleavage pattern. Various animal groups have notable, fundamental differences in these patterns, as the following examples demonstrate. Ctenophora, comb jellies. The first 2 cleavages are in the vertical, meridional, plane, figure 9.6a. The third division is nearly vertical and results in a curved plate of 8 larger cells, macromeres, figure 9.6b. The following division is horizontal and unequal, giving rise to small cells, micromeres, on the concave side of the tier of macromeres, figure 9.6c. The micromeres divide several times, figure 9.6d, and the macromeres produce a second set of micromeres, figure 9.6e. Mollusca, snails. Mollusk eggs exhibit spiral cleavage, in which each quartet of micromeres is rotated to the right or left of the macromeres in alternate divisions. The first 2 meridional divisions lead to formation of the A, B, C, and D cells. In the following cleavage, each of these cells, now called macromeres, divide to form a micromere. This process is repeated to produce further quartets of micromeres, which in turn produce further divisions, figure 9.7. Arthropoda, insecta. In the fruit fly, Drosophila, nuclei divide but remain in the same common cytoplasm, syncytium, figure 9.8. Gradually, the egg membrane folds inward to partition off each nucleus into single cells. This pattern of early syncytial development is true for most insects. Vertebrates. Cleavage patterns in the early embryo vary significantly between groups. Fish, frogs, chicks, and mice each have distinctive patterns of cell divisions, which are each distinct from the above-mentioned cleavage patterns. The crucial importance of early development is revealed by experiments in which individual cells from early cleavage stages are destroyed. In embryos such as mollusks, this leads to loss of adult structures descended from the deleted cells. Evolutionists have a tendency to downplay or overlook the significance of these early stages, but it should be emphasized that in such animals, the early stages of development really are foundational to the subsequent development of form. In some other animal groups, cell fate is established later. How then can fundamentally different early cleavage patterns be traced to a common ancestor? The proposed existence of such ancestors appears to be imposed on the evidence rather than emerging from it. 3.3 Cell fates. Tracing the fates of cells through development has enabled researchers to infer similarities, homologies, between organisms. For instance, by tracing mollusk embryo cell fate of different mollusks, it was found that the first quartet of micromeres eventually produces the head structures such as eyes, tentacles, and cerebral ganglia. The second and third quartet give rise to the shell, foot, and mantle cavity. According to embryologist Ivan Overkozas, this uniformity of cell fates is evidence that organisms sharing this pattern are related in some fundamental way. The prospective fates of cells in various regions of the embryo can be depicted as a so-called fate map, as can the distribution of gene products, which reveal fundamental discontinuities between basic forms, Figure 9.9. 3.4 Developmental mechanisms. Activities within the cell, such as behavior of its internal scaffolding, the cytoskeleton, can be unique to certain taxonomic groups. For example, in flies, the main body axis is established early in the egg before fertilization, when the cytoskeleton transports polarity-specifying gene products to opposite poles. In contrast, mammals define an initial axis by extruding tiny cells at one pole, but lack polarity components essential for development until at the 8-cell stage. Then, uniquely to mammals, dramatic cytoskeleton activity causes the cells to huddle together and flatten, compaction, and polarize along an inner-outer axis. Moreover, flies use a different mechanism from vertebrates to localize the machinery which orientates the chromosomes during cell division. Thus cleavage patterns, cell fates, and developmental mechanisms can be radically different between different forms of animals during these early stages. Davidson commented on how the various spatial patterns of embryos are generated. Some embryos begin this process by intercellular interaction, and others even before there are any cells that would carry out such interactions. Some rely on lineages that are committed to given functions as soon as they appear, others deal only in plastic, malleable cell fate assignments, some utilize eggs that before fertilization are cytoskeletally organized in both axes, some in one axis only, some apparently in neither. For some types of embryos every individual has a different cell lineage, whilst for others there is a set of rigidly reproducible canonical cell lineages, and some embryos display amazing regulative capacities. Thus, he concluded, the differences among the taxa in their modes of development are anything but trivial and superficial. 3.5 Hox genes and the phylotypic stage. Evolutionary embryologists commonly minimize these differences by speculating that patterns of genes, notably the body axis, specifying Hox gene cluster, show a common expression pattern in various animals, and that plasticity in such genes, particularly at the phylotypic stage, when members of a phylum might show maximum similarity, could generate evolutionary new body plans. However, there are several problems and flaws with these arguments. To reiterate a previous point, in numerous phyla, such as the mollusks, this supposed developmental plasticity at the phylotypic stage may be constrained by crucial earlier events in the embryo. Stages such as the gastrula, the proposed phylotypic stage of some invertebrates, could simply be a necessary developmental stage for these phyla, and an alternative explanation is that this is a common design pattern. The embryologist Michael Richardson demonstrated that there is no single phylotypic stage. This stage cannot be convincingly extended to the invertebrates, with cell fate maps and developmental mechanisms profoundly different between the various phyla when they are supposedly at their most similar. Flawed diagrammatic representation of the Hox clusters in research publications lead to misconceptions, according to Denis Duboul, professor of developmental genetics and genomics at both the École Polytechnique Fédérale and the Department of Genetics and Evolution at the University of Geneva. For instance, drawing genes as small boxes and the same size suggests that Hox genes are identical to one other, which Dubul states is rarely so. The exclusion of intergenic sequences is a biased perception that they are not important. For instance, two separate Hox complexes in Drosophila in this way become artificially juxtaposed, so that they align with their vertebrate counterparts, to give the misconception of their similarity. Dubul makes a surprising and embarrassing discovery and conclusion that vertebrates display the most tightly organized Hox gene clusters, whilst most other animals have a largely disorganized or split cluster, thus the textbook Hox cluster might be the exception and not the rule. This scenario would place vertebrates as direct descendants of the primitive ancestor from which the majority of animals are derived. Since this is so implausible, these discoveries would please the proponents of intelligent design. In other words, when the arrangements of Hox clusters between species are examined in detail, there is no transitional series toward the vertebrate pattern, which fails to show evidence of evolutionary links to a common ancestor. To reiterate, yet again, the morphogenetic action for the Hox genes, and their precise role in actually generating the phenotype from the genotype, is still unknown, and thus it remains highly speculative as to how Hox and other such genes could be generating evolutionary novel body plans. Indeed, evolutionists Rudolf Raff and Thomas Kaufmann admit a lack of evidence for evolutionary changes in body plans, commenting that most observations of evolution have not been made on organisms, but on fossils, for only by recourse to the fossil record do we gain a view of organisms' actual evolutionary histories. 3.6 The fossil record. So what are Raff and Kaufmann's observations of evolution within the fossil record? The Cambrian explosion refers to the dramatic appearance of most of the animal phyla at the base of the hard-bodied fossil geological record, and is global in its extent. Raff and Kaufmann make a number of surprising observations. Highly complex animals, echinoderms, trilobites and arthropods and several classes of mollusks. All appeared in the Cambrian period in considerable diversity and without recognized ancestors. They note that echinoderms, example, starfish, for instance, appear in the Cambrian with a lack of any identifiable ancestors, appearing with all the basic echinoderm patterns fully recognizable. That is to say, the fossils in the Cambrian can be identified with the forms of their modern-day counterparts. Transitional forms are largely hypothetical, even the earliest echinoderm classes are quite distinct from one another. This data is not restricted to echinoderms, the gaps are general and prevalent throughout the fossil record. New morphological structures do appear, example, the amphibian limb, wings, but when they first appear in the fossil record they are fully formed, and are not transitional. How the various multicellular body plans arose and diverged from a common ancestor have proved to be fertile grounds for speculation, because there are so few facts to restrain the imagination. This is an extraordinary admission from such eminent evolutionists, and reiterates the speculative scenarios of the Evo-Devo community. Convincing evidence in strata earlier than the Cambrian for the origin of these diverse phyla is lacking, and attempts to spread out the Cambrian period, implying that the first appearances were less abrupt, have been thwarted by new examples of fossils appearing that push the timescales back again. Efforts to ascribe the right geochemical conditions to be a trigger for the Cambrian explosion are also unconvincing, because the chemical environment provides merely the permissive conditions, not the cause of complex specified information embodied in the Cambrian body forms. From my boat-building experiments, I now have a collection of failed prototypes, such as boats that are too unstable. But in the fossil record, where are the failed prototypes? They are nowhere to be found. Instead we find nature got it right from their first appearances, as in the perfectly balanced cantilever bridge structures, figure 9.10a, of dinosaur skeletons such as sauropods, with their massive tails perfectly counterweighting the equally massive, long neck. The two balanced cantilevers are bound up in one common field of force and plan of construction, in which the stress diagram, representing all the forces acting on the animal, becomes the structural plan. For instance, in Brontosaurus, figure 9.10b, and Apatosaurus, figure 9.10c, the backbone cantilever arch culminates over the pelvis and hind limbs, and the highest vertebral spines are found there, to dissipate the maximum tension loads experienced in this region, and with the height of the spines grading perfectly in proportion to the forces experienced along the length of the backbone. Without this, the bones would break under the enormous weight of the animal. Moreover, dinosaur skeletons are not merely like static bridges, but show a capacity to modify their form in order to bear the loads during dynamic motion, figure 9.10d. Huge fossil graveyards can be seen today, but they are not graveyards of failed design. 3.7 Genetic toolkits. Evolutionary geneticists require mutations in early development to produce large-scale changes in the body form, because in later development the body plan is already established. However, studies have revealed that groups considered the most primitive, example, sponges and jellyfish, are found to possess most of the genetic toolkit, such as 11 of the 12 WNT gene families, various signaling pathways, and even molecular organizing centers, that are also found in the so-called more advanced vertebrates. This is evidence against gene diversification underlying the so-called Cambrian explosion. It is also being discovered that genes expressed during early embryogenesis involve more interactions than those expressed during later stages, suggesting that they are actually less malleable to transformation at these stages. 3.8 Taxonomic comparison of gene expression. There are evidences of peculiarities in various gene expression patterns between taxonomic groups. For instance, the patterns involved in leg development differ between insects and spiders. Many transcription factors found in amphibian eye development do not operate in mammals, along with other considerable differences between vertebrates. In conclusion, the above data provide an evidence base for the existence of types that are real in nature. Distinct forms are apparent at various hierarchical levels, ranging from the phylum level, representing the basic body plan, down to the basic types, most commonly identifiable with the family level, and recognizably distinguishable from the nearest forms by clear gaps or discontinuities. This evidence-based approach reveals that modern evolutionary theory is struggling to engage with the real world, and that much of the discussion remains highly speculative and driven by the presupposition that the diversity of life must be explained by a universal common ancestor acted on by blind natural processes. The challenge for theistic evolutionists is whether they want to align themselves with speculative naturalism, or whether they are willing to follow the evidences for design and for discontinuity wherever this leads. Concluding remarks. In Buchan's novels, the characters spectacularly encounter one another at just the right time and place, because the plot crucially depends on it. Without Sir Archie and his biplane, Hanoi will be arrested and the spymaster's schemes will not be thwarted, influencing the outcome of the war. In similar vein, in development the cell and molecular characters and forces encounter one another at just the right time and place, because the progress of embryological development crucially depends on it. This is evident, for instance, in the placement of genes and skeleton-forming cells precisely to future joint locations, the connecting of a particular motor neuron with its correct target muscle, the complex interplay of transcription factors during heart development, which is yet synchronous with the orderly development of the circulatory system, and the electric current appearing at the exact location to mark the future limb bud. Evidences of such orchestration are growing in abundance, and a new language is emerging, with secular authors even adding superlatives: it is exquisitely orchestrated. It is increasingly untenable to reconcile this as emerging from Darwinian chance processes, or even by the spontaneous self-assembly of organisms according to the laws of physics and chemistry. To explain the story without recourse to a directing intelligent agency seems increasingly far too far-fetched. Wernicke comments that, as engineers realize, all of organisms' parts are in place and functional. This is only achieved if there is a higher-level instructional program that orchestrates the operation of every subsystem in the overall system. Constructing these instructional programs requires complete knowledge of every subsystem as well as complete knowledge of the entire system before the higher-level program can be developed, implemented, and accomplish its goal. For human vision, for instance, it is inconceivable that these components would just randomly integrate themselves to operate as a single harmonious unit that provides sight. Furthermore, structures such as wings are not simple and flat but develop into marvels of engineering, ranging from efficient hovering, to gliding, or fast-forward flight. These have far-reaching implications for development of the entire body plan to suit the type of flight mode. For instance, in very fast flyers such as swifts, the wings are swept back for high-speed flight, then sweep outwards to effect tight turns, figure 9.11a. This is mimicked in the design of variable wing fighter planes and micro-air vehicles, MAVs, figure 9.11b and c. The Swift is much more efficient than these planes because it continuously adjusts, morphs, its wings for maneuvers. In labs worldwide, intense biomechanics research into the intricacies of bird and insect flight, Figure 9.11d, aims to inform state-of-the-art aeronautics design. For instance, it is being discovered that many bird and insect wings have a sharp leading edge which creates a vortex, a tiny tornado, that would be expected to contribute to stalling but instead sucks in air to generate extra lift, Figure 9.11e, and aids aerobatics and landing, say, on a branch, without the animal losing altitude. In Buchan's novels, Hanoi and his little band of comrades have instructions and clues to follow from their commanding officer. There are many possibilities as they initially speculate over these clues. But then they find the lines of evidence which best fit the clues. So it is with development. Evolutionary theory predicts chance processes and tinkering of imperfect assemblies. However, the evidence from development indicates the necessity of a designing intelligence. In my mind, this points to the creator who orchestrated the intricate construction of each distinct type of body form found in nature.